are there, and in the afternoon session we have one practical is there. And now it is a time for introduction of the germplasm management classes. Very good lecture, highly informative lecture. And let me introduce our current speaker. She is Dr. Anjalika. She is a principal scientist and head division of germplasm conservation in NBPGR. And she is a gene bank creator and expertise in conservation of fiber and forage genetic resources, and team leader in management of information in the National Germplasm Conservation Network. And she done a pioneer work in conventional seed storage methods of conservation of fiber and forage germplasm, especially in wild hibiscus species. And she is since long time she is working on registration of plant crop germplasm, and she developed inventories, and every year she is making the inventories. And currently she is looking all the activities of germplasm management, as she is a head of and National Gene Bank. It is located, and it is a second world gene bank, biggest. And she is a fellow of Indian Society of Plant Genetic Resources, and she is a reviewer in several international reputed journals. And she registered genetic stocks in cotton for novel traits, and nearly 6 noble traits. And her publications are research papers nearly 48, in other books, bulletins, and annual reports, technical proceedings, training manuals, and some are beyond this. All she involved in actively in all scientific committees in the institute and other important committees. And let us invite our speaker, ma'am. We are welcoming you and for delivering your lectures, and please kindly share your expertise and knowledge to our participants. The participants are nearly 84 participants are there, ma'am. They are from 18 organizations. And please, ma'am, already participants are requested. They have more doubts in germplasm conservation. Please kindly deliver your presentation, and please continue, ma'am.
It will be in an interactive mode. Whatever questions they have, they can ask me during the presentation itself if they have some queries.
Yes, ma'am.
You can answer.
You can interact with ma'am also, and she is very friendly, and she is having a vast knowledge of experience. And please, ma'am.
Thank you, Ravi.
Thank you.
So we will start with the presentation.
Yes, ma'am. Share screen, ma'am. Yes. Yes, ma'am. It is visible.
You are able to see? Am I audible also?
Yes, ma'am.
Okay, then should I start?
Yes, ma'am.
Good morning, participants. The topic of today's lecture is gene bank management and ex situ conservation. So before we proceed for the lecture, we should know some basic definitions we should use. So first of all, what is PGR? Actually, PGR are the genes which are of direct or indirect value to the human beings. And actually, first slide is this. So what are the genetic resources? Genetic resources, actually, these are the genes of present and potential value to the human being. So this is the genetic resources as per the CBD. Now, plant genetic resources includes all the agricultural crops. Their wild relatives comprises of land races, farmers' varieties of seed cultivars, breeding lines, and potential domesticates or wild species. Now, germplasm. We refer to germplasm many a time. But what is germplasm? Germplasm basically refers to the reproductive or the vegetative propagating material such as seeds, tissue, cell, pollen, DNA, which contains the functional unit of heredity and can be used in the crop improvement program. So these sometimes for every reproductive or vegetative propagule which we use for conservation and has a functional unit of heredity is germplasm. So starting with this. Now, why there is a need to conserve the PGR? As you all know that lots of release varieties are developed. They have a very narrow genetic base, and we go in for the these monoculture of these improved cultivars, and this has led to the loss of the genetic variability And other things like industrialization, urbanization, and development has also led to the genetic erosion and extinction of these species. And these species, this PGR is a very valuable raw material for the crop improvement programs, which is very important for our food and nutritional security. And PGR is a national heritage, which we need to manage and conserve for the posterity. Because you know that even if we use all the biotechnological tools, it is not possible to recreate each and every variation or the diversity in the germplasm. Even if we are able to do it, it's a very costly proportion. So it's better to conserve the genetic resources as they are available in the nature. So this is why we need to conserve the plant genetic resources. Now, there are conservation strategies for plant genetic resources for food and agriculture. There are two conservation strategies made. One is, these are as per the definitions as per the CBD, that is CBD 1992. Ex situ means when you conserve the plant genetic resources outside their natural habitat. So you conserve it in a research institute, you conserve it in a gene bank. Even if you conserve it in a field gene bank, it just means ex situ conservation. You are not conserving it where it is, where the germplasm itself is. So you are taking it from there, that place outside and conserving it in an institute or a gene bank or a field gene bank. That is ex situ conservation. Now, in situ conservation means that you are conserving the ecosystems, the natural habitat, so that the viable populations of the species can be maintained or have to recover in their natural surroundings. So in case of domesticated or cultivated species, these are the surroundings where they develop their distinctive properties. So these are the definitions as per the CBD. Now, there are various strategies. There are two major strategies, as I told you, in situ and the ex situ. In situ means in the original habitat, and ex situ means outside the original habitat. So in situ this conservation is a very dynamic method, because in this you allow for the evolutionary process to go on. Like biosecure reserves, we have national parks, we have gene sanctuaries, on-farm conservation. So these are all the types of in situ strategies for conserving the this plant genetic resources. However, ex situ conservation means when you are taking them outside their natural habitat. So these include, this is however a static strategy of conserving the germplasm. Static means that you are not allowing for the evolutionary process. You are just conserving as it is. So there are various strategies and types of this ex situ conservation, that is field gene banks. So field gene banks are also conserved ex situ conservation strategies, because you are bringing the germplasm from other places and are maintaining them in a gene bank, in a field gene bank. Then we have the seed gene bank, in vitro gene bank, cryo gene bank, and the DNA gene bank. These are all the types of ex situ conservation. Next is the history of the gene bank. So here mostly we will be concentrating, or our lecture is mostly on the seed gene banks. So this is the history of the gene banks. All the gene banks, there are many gene banks in the world, but the apex organization for the global ex situ conservation are gene banks of the consultative group on international agriculture and research. So this is based on a signed agreement between FAO in 1994. CGIAR gene bank started conservation. That is CGIAR gene banks तो 11 है, जैसे AD है, ICADA है, ILRI है, ITA है, AVRGC है. So these are some of the gene banks that come under this consultative. Even ICRISAT is a consultative group of international agriculture research के अंदर आता है. And these gene bank conserve genetic resources in trust. What do you mean by in trust? In trust means that they conserve the gene bank for all the countries, but they do not have an intellectual property right of the germplasm. So they have to give the germplasm or share the germplasm based on the international regulations for the exchange of germplasm. That means in trust. In germplasm conservation, we use two terms: in trust, black box arrangement. So here the CGIAR gene banks, they conserve the germplasm in trust. In trust means that they will be having any intellectual property right over the germplasm they conserved in their gene banks. So global gene banks, there are about 1750 individual gene banks in the world, and 8 have more than 1 lakh accessions and 130 hold more than 10,000 accessions. So CGR IRI gene banks form a very important component of this ex situ conservation. Now, what are the advantages and the disadvantages of a seed gene bank? Actually, conserving this this germplasm in the form of seeds is very easy, and it is less costlier than other methods. So you can have a large number of accessions. Even entire variability can be conserved in a very small space. In seed banks, the germplasm handling is easy, and you can conserve it under a pathogen and insect-free environment. So these are the advantages of the seed gene bank, and it is cost-effective also. However, the disadvantages are that since we have recalcitrant species that cannot be stored in the gene banks, and power failure supply may sometimes lead to loss of viability. And when we conserve in the gene banks, we need to have the periodical evaluation of the seed viability, and multiplication is also very important. So these are some of the disadvantages of the gene banks, but the advantages are more than the disadvantages. So most preferred conservation method is conservation in the form of a seed, especially in case of orthodox seeds. As you know that orthodox seeds form a major constituent of most of the plants have orthodox seeds. More than 90-95% plants or the germplasm have orthodox seeds. So conservation in the form of seed gene banks is the most effective one. Now, according to international gene bank databases that are available in the public domain, there are the four largest gene banks at national gene banks are the National Center for Genetic Resource Preservation in the United States of America. Then next is our gene bank, that is the National Bureau of Plant Genetic Resources. It's located in New Delhi, and it was established in the year 1986. New facility which we have developed in the new campus was inaugurated in 1996, and then we did the modernization. Last year, we brought new components of modernization like biometric for each and every module, etc. So that modernization was done with the gene bank. So third gene bank is the Chinese Academy Gene Bank, and the fourth one is the Revlo Gene Bank in Russia. So these are the four largest international gene banks, and our gene bank is on the second number. So this is located in NBPGR. We have a network of 10 regional stations. Basically, the point of having the regional stations is to have the stations in different agroclimatic zones, so that we can collect the material in the vicinity of those these regional stations. Exploration can be taken in those, they can undertake the exploration in these areas, and can also help in the characterization and maintenance of that germplasm, because it is the conditions there are same to the conditions of the germplasm that has been collected from these areas. We also have 59 national active germplasm sites, because we deal with a large number of species, and it is not often possible for the regional stations to take all the activities. So for that, we have these crop-based institutes. So if we want to characterize, evaluate, multiply, and that is a responsibility of these crop-based institutes, because they have expertise for each and every crop. Maybe in NBPGR, we may not be having expertise for each and every crop. So we have the linkages with the national active germplasm sites. So this is the entire national germplasm network. So here you can see that we have an in vitro gene bank. We have a cryogenic gene bank, which maintains the cryotanks are maintained at about -180 to -196. We have 6 cryotanks. We have field gene banks in here itself in Delhi, and in our Isapur farm, and also at our regional stations. Then in the seed gene bank, we have a medium-term module. We have 5 medium-term modules which maintain a temperature of 4 to 8 degrees centigrade. And then we have a long-term module that is maintained at -18 to -20. So we have 12 such modules. Then medium-term modules are also available at the NAG sites also. Some of them are maintaining, others there is a problem in maintaining the medium-term modules also, because it is a very costly affair. A lot of electricity is there, and sometimes the expertise is not available. So technical support is not available. Because of that, running the medium-term modules itself is a very difficult this process. But we have 5 in our national gene bank. Now, the types of storage in the gene bank. So, there are major three types of storages. There can be three types of storages in a gene bank. So, storage, short-term storage is 12 to 18 months. So, this type of storage we don't maintain at NBPGR. This type of storage is normally maintained by the breeders who want their material for from one season to another season. That's why the storage period is 12 to 18 months. And the temperature is 20 to 22 degrees centigrade. That means if we install a that's if we install a AC in a room, it is as good as a short-term storage facility. So the container they are used are cloth bags, paper bags, glass bottles. So medium-term storage and long-term storage is what we maintain in the seed gene bank. So medium-term storage means storing a sample for 5 to 10 years. Temperature could vary anywhere between 0 to 10 degrees centigrade, or it has to be maintained at 20 to 30. And the containers are cloth bags, paper bags, metal cans, glass bottles, and plastic jars. From medium term storage is mostly meant for the active collections. Active collections means wherefrom you have to distribute the material, or you keep some material which doesn't have some issues related to passport data, or some other issue, or you have less seed quantity. That type of the material goes into the medium term. Since we invest a lot of public money in the explorations and collection of the material, suppose the material does not qualify for the gene bank standards due to certain reasons like low viability or less seed quantity, so we cannot just throw the material, because we have invested so much of money into bringing that material into the gene bank. So we will keep it in the medium term. Then when we find some suitable person or a suitable organization where we can send that material, so in the meantime, we keep the material in the medium term. So medium term is mostly material in the transit. So here you can conserve it for 5 to 10 years, and the temperature of medium term facility varies from 0 to 10 degrees centigrade. Here at NBPGR, we maintain at 4 to 8. And RH has to be maintained at 20 to 30%. Since you are keeping the samples in cloth bags, paper bags, sometimes some people prefer to keep it in glass bottles or metal cans, but cloth bags and paper packets is a better option, because there the exchange of moisture, this gases takes place, and the seeds are maintained at a low moisture content. And then third facility is the long-term facility. Long-term facility, the storage period is 50 to 100 years. And the temperature here is maintained at -80 to -20 degrees centigrade. We don't need to control the RH. Now here, no need to control the RH, because we are already sealing the packets in tri-layered aluminum foil pouches. So there is no need to maintain the RH. However, the temperature has to be maintained at -18 to -20. So here in the national gene bank, we are maintaining at -18, because by 2 degrees this variation in temperature, we can economize on the electricity, and there is not added benefit by reducing the moisture by 2 degrees. So we maintain mostly at -18 degrees centigrade. So these are the types of storage that are in the gene bank, but short-term facility we don't have here. Medium and long-term we have. So medium term we have 5 modules, and long-term we have 12 modules. Now, these are the global comparison of the global gene bank versus the Indian gene bank. Here you can see that we have 4,63,130 accessions, and the number of species is 2,108. Here it is interesting to know that Millennium Gene Bank has 80,000 accessions, but the number of species is 37,600. It is because that we are conserving mostly the agrobiodiversity, where they are conserving the entire biodiversity in the Millennium Seed Gene Bank. That is why the number of species is more. Since our mandate is agrobiodiversity, so we will be conserving mostly the agriculturally important species, along with their crop wild relatives and their BD relatives, wild and BD relatives, crop wild relatives. So the number of species in our gene bank is less, that is 2,108. But the number of accessions, you can see we have 463,130. So in this gene bank itself, the number of cereal crops is more, that is 1,73,487. Because the number of accessions in rice, we have 1,16,000 accessions in rice itself. because rice is a crop indigenous to India. So we have a lot of diversity in that crop. So the number of accessions obviously the number of accessions in the paddy crop is more, that is more like 16,000. Likewise, you can see in other crop groups like millets, forages, we have collections. So maximum we have in these oil seeds, grain legumes, since they are the major crop groups, the number of accessions in this is high. Now, it depends upon how to select a suitable conservation method. Now it depends what you want to conserve. It depends upon many things, that which will be the suitable conservation method, whether we should conserve it in a long term, whether we should conserve it in medium term, should we conserve it in a field gene bank. So that depends upon many things like infrastructure and technical expertise. Suppose you don't have that much infrastructure, you may not go for a long-term conservation, you would like to have conserve in the medium term. And technical expertise for cryopreservation is not available, so you may go directly for the seed conservation. So there are many things on which the suitable method for conservation depends. But one of the major factors on which the conservation depends is the nature of the propagule. So it depends mostly on the nature of the propagule if other things are taken care of. So we should know about the seed storage philosophy. Suppose we cannot conserve mango in a gene bank, mango seed in a gene bank. We cannot conserve rambutan seed, or we cannot conserve coffee in a seed gene bank. So we have to understand the seed storage physiology first. So mainly there are based on the seed storage behavior, there are three different types of seeds. That is the orthodox, the intermediate, and the recalcitrant seed. Orthodox seeds mean they are able to survive long time dry storage. Intermediates are they can withstand dehydration to some extent, and recalcitrant seeds are those seeds which cannot withstand dehydration. So it is based on these physiological aspects of the seed storage physiology that defines that which type of the conservation strategy we can use. Normally, orthodox seeds they can undergo maturation drying. They are shed at a dry condition that is less than 20 moisture content. But intermediate and recalcitrant seeds, they have absence of maturation drying, and they shed the seeds at more than 50% moisture content. So orthodox seeds can be dried to a level of less than 7%, whereas intermediate can be dried to 7 to 18%, and recalcitrant seeds we cannot dry below -20. So if you cannot dry it below -20, obviously you cannot conserve it at -18, because there will be freeze injury to the seed. So these types of seeds can be conserved in the allen. So we'll have to go for the cryo for these recalcitrant type of seeds. But for orthodox or intermediate seeds can be conserved in sub-zero temperatures for some short duration. And these orthodox seeds, which constitute the major chunk of the seeds, they can be stored in for many years at ambient, low, and ultra low moisture conditions. So how we will determine that whether the seed is an orthodox, intermediate, or recalcitrant? Suppose when you receive a seed, you try to dry it to a moisture content of 10 to 12%. So if most of the seeds die, it is a recalcitrant seed. So then after that, you dry it again. If if you have brought it to a moisture content of 10 to 12%, you dry it. If they survive the viability test, you can dry it further to 5%. And then again do the viability testing. If most of the seeds die, that means it is an intermediate type of a seed. Then again, you store these seeds at 5% moisture content. You store this for -20 for 3 months and undertake the viability test again. If more seeds die, it is an intermediate type of a seed. And if almost all the seeds survive, so it is an orthodox seed. So orthodox seeds are basically those seeds which can tolerate desiccation up to a moisture content of maybe say 5 to 7%. Then we can safely conserve it in the seed gene bank. Now, this is actually the operational sequence of the integration of the accession in the national gene bank. So when you send a seed sample, this is the flow chart, how we proceed. So first, when we acquire the seed sample, we verify the documents. So documents are to be verified. What are the documents that you have to send? First is the passport data in case of germplasm. Suppose you have collected a sample and you want to submit it in the gene bank for a national identity and conservation. So passport data you have to, passport data sheet you have to fill. In the passport data sheet, there are various columns. I will show you it at the end of the presentation. So passport data means where from you have collected, what is the longitude, latitude, altitude of that, whether what was the habitat where from it you have collected, whether it was wild, whether it was VD. So those all those things have to be written because we should know without passport data, it is like a junk you are conserving in the gene bank. So if in case of release variety, suppose now you have released you have a variety which you want to release with CVRC or state and you want to conserve the germplasm because it is mandatory. So in those cases you will have to send the proposal of the release varieties. So along with the sample, send the proposal of the release variety. Then in case you want to register a trait-specific germplasm in NBBGR, suppose you have some germplasm for biotic stress, tolerant to certain drought, or maybe even high yielding, some unique type of a germplasm, and you want to register it with a trait. In that case, you will have to fill in a registration profile. On that, there will be another lecture with Dr. Neena Gupta. So she will tell you in detail about the registration of germplasm. So in that case also, you will have to send the required quantity of the seed, recommended quantity of the seed, and you will have to fill in the registration proforma, which is online. So three types of verification documents you have to submit the seed if you are submitting the seed. One is passport data sheet, proposals for release varieties, and registration proposal in case of registered germplasm. And if all these documents are complete, if not, we will send it back to the donor and get the information or instruct them how to fill in the forms or how the data sheet is to be filled. Even those things can be, NBPGR can help in those things also. If you have to fill in a registration profile or you are not able to fill it, those things we can help you. So if all the documents are complete, then we have to send this to a domestic quarantine. Since the samples come along with some weed seeds, some other seed material, or they may be having certain pathogens in that, so we have to send it for a domestic quarantine. So once the center and the seed sample we receive it in the germplasm handling database. There we check for the duplicates because redundancy in gene banks is one of the major issues. You can see that for a single accession, we may be having 10 duplicates, which adds to the burden of the gene bank. So checking the redundancy is one of the things we do in the GH2, germplasm handling unit. We check that if this sample or if this landrace is already available or not. So we have to check for the redundancy also. So once the sample is unique and the quantity of the seed is sufficient, we after getting it quarantined, we clean the sample, etc., remove these weed seeds, some soil samples or something is inside that. And if there is some some brucates, etc., we can even fumigate the sample. The quarantine people will fumigate, and the fumigated sample will be sent back to the donor. But rest of the samples, if they are clean and there is no pest, etc., in it, and one thing people should always know while sending a sample to the gene bank is that we should not treat the seeds. It should be untreated seeds. Fully physiologically mature seeds, untreated seeds should be sent. Sometimes they will just have some coloring and all other or fungicide treated or with naphthalene ball. These things should be avoided. You should send the seeds which is physiologically mature within 60 days of its harvest immediately for the conservation purpose. So after this health check of the seeds is done, the material is sent to the germplasm is actually sent to the crop curator where he does the viability testing. So most of the seeds we should have viability test. We should have viability more than 85%. But this has to be relaxed because suppose in case of cotton, this certification standard itself is 65. Or in case of grasses, sometimes we have to relax to 20% also because there is a lot of fuzzy, these awns and all other material in the seed, this grass seed. So in that case, it is relaxed to even 20. In sugarcane also we have relaxed it to 20. because we have this true seed of sugarcane also conserved in the gene bank. So in some cases and wild relatives, we can relax the standard to some extent. Like in vegetables, it is not possible sometimes to have 2000 seeds. Like we have this gene bank standard is to have 2000 seeds for self-pollinated and 4000 for the cross-pollinated. So sometimes it is not possible to have that much seed quantity in case of wild species or in case of difficult to seed species. In that case to case basis, the curator can relax the standards. So once the all procedure is complete, the material is sent to the gene bank after drying, etc. Once it is dried, we will assign an IC number to this indigenous collection number to the germplasm, and once it is done, it is communicated to the developer or the collector, whosoever has collected the material. So these are the seed gene bank procedure. First method is cleaning. You have to clean the material because your pure sample of the seed of highly physiological maturity has to be stored. Then moisture testing is done as per the seed testing the ISTA rules. And seed viability, we use two methods for seed viability. That is the top of the paper method and between the paper method. Top of the paper method means that if you have small seeds, you will do it in a petri plate. But if you have large seeds, so large seeds cannot be done in a petri plate. So those are done in the between the paper method in paper towels. That is the most of the people who do seed technology work, they must be knowing that what's the difference between the top of the paper, and maybe in practicals also they will show. Then in case the seeds are dormant, we have to go for the tetrazolium testing. Tetrazolium testing means that we keep the seed in the, we hydrate the seed first and then do the tetrazolium testing. You can here see that the embryos turn pink. So that means the seed is viable. So we go for the tetrazolium testing in case of dormant seeds to see that if they are viable or not, then we conserve the seeds. Because every time it is not possible in a gene bank to go in for the dormancy breaking treatments, etc., because the quantity of the seed is very less. So the next procedure is drying of the seed. So now that the seed has been processed, everything has been done, we have to dry these seeds. So they are dried in these dryers where the RH remains 15% and the temperature is 15 degrees centigrade. So this is the safe method of drying. So it is recommended if somebody is sending a sample to the gene bank, they should not dry it in sun. It has to be bench dried. You can dry it in a lab. After drying, bring in the temperature to maybe 10 to 12% even. You can send it for the gene bank. So there we will reduce the moisture content to 5 to 7% in these dryers, which have a RH of 15% and temperature at 15 degrees centigrade. So at high temperatures and high RH, we should not try to dry the seed because it reduces the viability of the seed. So once the drying process is complete, we will pack them in the tri-layered aluminum foil pouches. So these are the most effective way of conserving because you can cut the sample anytime you want to take out this material and it is very easy to handle. So packaging material used in the gene bank is these tri-layered aluminum foil pouches. They have 3 layers. One is the outer polyester layer, which is very essential for the packing of the seeds. Then we have the middle aluminum layer, which is moisture impervious for moisture impervious, and inner polythene layer that is to keep the sample sturdy. Since you have to maintain the sample for a very long period of time in the under -18 conditions, so it has to be very thick and of a very good quality. So this storage I have already told you. Now, once everything has been done, the processing of the material is complete. All the information should be in the database because the person should know what is exactly in the gene bank. So all the information related to the passport data and related to the gene bank data, like moisture, what's the moisture of the sample, what's the number of the seed quantity, and number of seeds stored, the germination percentage or viability of the seeds, and all the passport data is entered into the database. That is the germplasm information management system. So here you can have all the reports like passport report, what is the passport data of the, you can have gene bank reports. So what is the next date for monitoring, whether the material is available in the gene bank. So all this information can be had from the database. So we enter all the information in the database. This helps in locating the material in the gene bank, and also we can keep a track of the monitoring, availability, and all other things. Now, once after the conservation of the gene bank, in the gene bank, it is very important that we maintain a high proportion of the viable seed under optimum conditions and periodic monitoring of the seeds for viability and quantity and regenerating them when the situation warrants. So we have to manage these seeds in the gene bank. So if we are keeping the seeds in the active collection or in the MTS, we have to monitor every 5 to 3 years, depending upon the nature of the seed. If it is a non-oil rich seed, you can monitor it every 5 years. And if it is an oil rich seed, you can monitor it in 3 years. But in the base collection, that is -18 or -20, you can monitor non-oil rich seeds after every 10 years and the oil rich seeds after 5 years. Now, this depends upon the initial viability also. So you can just change the monitoring intervals depending upon the viability. If the viability of the seed is more, say more than 95%, you can increase the monitoring interval, like 20 years for non-oily seeds and 10 years for the oily seeds. But the most interval we are using is 10 to 5. Normally we use 10 or for 10 years for oil seed rich seeds and 5 years, every 5 years for oil rich seeds, because oil seeds are slightly poor storers. But so far we have been doing monitoring for a 20 or 30-year-old seeds. Now that our gene bank is approaching 30-year storage this period, we have observed that there is not much loss of viability. The samples are maintaining their viability. So we are monitoring over a period of 10, 20, 30 years. We have 30-year-old samples also. So we are monitoring them. There is not much loss, maybe in some oil seeds. So in that case, we have to go for regeneration. If the this germination drops below 85, we have to go for regeneration. That is for where the germination is more than 85. But in crops where it is 65, that there also, if the germination goes down by 5%, we have to go for regeneration of the samples. So regeneration should be done. Adequate number of the plants should be grown so as to minimize the genetic drift. And during regeneration, in order to maintain the genetic integrity in cross-pollinating crops, they should be well maintained by pollination control, so that there is not mixing of the lines. Now, these are the seed gene bank standards. These are the seed gene bank standards in a gist. First is that if you have to submit a seed sample for the gene bank, you should have untreated seed material. You should not treat, you should not even put in some fungicide or naphthalene balls. Nothing should be put inside the seed. And it should be packed in maybe in a paper packet or a muslin cloth bag rather than a polythene bag. Because if you put it in a polythene bag, if the moisture content of the seed is high, it will lose its viability during transit. So it is better to keep it in this aluminum with this paper packet or a muslin cloth bag. That is the best way to send the seed. Now the seeds should be of high quality with maximum viability, that is 85%. But in wild species, in difficult species like vegetable crops, the standards can be relaxed. Even in cotton, it is 65%. And depending upon the pollination behavior of the seed, 2,000 seeds have to be supplied for self-pollinated and 4,000 seeds for the cross-pollinated. And moisture content, you don't have to worry about the moisture content. If it is 10 to 12, it's okay, because we are going to, anyway, we are going to reduce it to 5 to 7%. And then seed storage temperature is -18 if it qualifies for the long term. If it doesn't qualify, then we keep it in the medium term storage. And the storage monitoring viability after 10 years in long term, 5 years in medium term, and for regeneration, 10 or more plants are generated to avoid the loss of it. So these are in just the seed gene bank standards. So if you have to supply the seed, you should know that how much of the seed has to be supplied, what should be the viability and moisture content of the seed, and when you are supplying the seed for the gene bank. Now, another issue related to the gene banks is that we need to have a safety copy gene bank. There is forward gene bank, so we also need to have a safety copy gene bank, because you can see that in Syria there was a civil war and all the facilities were fell to the rebels, and it was they had done one thing was that they had submitted the material in the Svalbard gene bank. Even after this, all this the gene bank was destroyed and all other things. They were able to retrieve the material, and that material from the Svalbard was then grown in the Lebanon and Morocco, where we have now again the germplasm is safe. So, in order to mitigate the risk of partial or total loss caused by the natural or man-made catastrophes, we should have a safety copy gene bank. It is as per the FAO recommendations. So in the safety copy gene banks, a black box approach is followed, generally followed. Black box approach means that the germplasm can be accessed only by the country which has deposited the material. The gene bank itself doesn't have any right to distribute the material. So if somebody wants the material from the Svalbard gene bank, they cannot get it because only the country, it is actually a safety security type thing you have kept in a vault in the in Svalbard. So if I have submitted the material, our gene bank has submitted the material, we can only open the material. There it remains in a black box type of arrangement. That means no other person can deal with that material. So there are certain criteria where a safety gene bank can be constructed. It should have low proper seismic activity, low homogeneity, low radioactivity. It should have a good infrastructure, support good accessibility, and there should be low risk of political embargo or terrorism and low risk of flooding. And overall operational expenses should be less. So this is the criteria for the geographic location and the environmental conditions where we can have a safety duplicate gene bank. So India is also in the process of having a safety duplicate gene bank. We have surveyed many areas in Himachal and in Uttarakhand. We had also selected one location in Ladakh, but then after that the Doklam happened and we couldn't continue with that. So these are the criteria for geographic location for the this safety duplicate gene bank. So this Svalbard gene bank was opened in 2008 and it was funded by the Norwegian government. And today you can just see the structure of the Svalbard gene bank. It has a tunnel right from the, you can carry on the trolleys, the seeds can be carried on the trolleys. It has 3 vault rooms and this one is occupied. Here we have the seeds. So it is a permanent frost condition here. So the operational cost here is low and there is a chance of low seismic activity here also. So people are conserving in the Svalbard gene bank. Now, as far as India is concerned, you can see that our director, Dr. Bansal, had been to Svalbard gene bank for submitting the seeds. So initially, we submitted some 5 seeds, 5 seed samples, 2 of rice and 3 of wheat. After that, pigeon P25, then 100 of sorghum and 100 rice in 2017, and finally in 2020, we submitted the 4 sets of wheat and chickpea to the Svalbard gene bank. So they will be as a safety duplicate there as and when we require them. We can just access the material since it is in a black box arrangement. So no other country has any right on this material. Now, these are all the things which we conserve in the gene bank, that is land races, improved cultivars, wild and weedy relatives, genetic stocks, release varieties, parental lines, farmers varieties. This is all what we conserve in the gene bank. Now, this is the passport data sheet. I think exploration people might have already told you about these things, but people have more confusion about this biological status. So what is the biological status of the seed sample? Here only people will do a large number of mistakes. So I will just tell you that what is this. These are the descriptive states of the state of definition. So wild means all the species other than cultivated. That means wheat, rice, you can consider a cultivated, Triticum aestivum, Triticum durum, which is a cultivated variety. But all other species of Triticum will be wild species. Then crop wild relatives will be related to the cultivated species. All those species that are in the gene pool of the cultivated species. So that will be crop wild relatives. So you will have to write, if you have a wild species, any wild species, that you will write as WF. All crop wild relatives you will write as WL, CWR, and VD relative. VD means that a cultivated species which is in companionship with the other cultivated species. Suppose we have a mustard plant in a field of wheat. So it is a weedy species there. So you can write weed. If you have collected it as a weed from some cultivated field, it will be a weed. The biological status will be weedy. Likewise, we have land races. So land races are normally the cultivars that are found in the farmer's field. They may be having a name or they may be without name. They have some unique traits. So LR you can write, or you may be having some traditional or primitive cultivars like Sikkim primitive is a land race which has been in use during the primitive times, but ancient times, but now it is not in use. So it is a traditional cultivar or a primitive cultivar. Likewise, we have farmers variety. Those are the varieties, extent of the obsolete varieties that are registered with PPVFRA. So these varieties also can be conserved in the gene bank. So for that, the biological status will be approved. Then all the breeding material, that is semi-finished products or segregating material generated out of a breeding program to meet the specific breeding objective. So in this, you can have anything. Suppose you are not able to distinguish between the two, you can simply write BR, breeding material. So in breeding material, we have hybrids, inbreds, segregating population, breeding lines, parents of hybrids. Then we have the genetic stock. Genetic stock means that through some human intervention, you have developed some trait-specific germplasm that has some unique trait. So that is a genetic stock. Then registered genetic stock means the genetic stock which you have registered with NBPGR. That will be GSRG. So this genetic stock we will be adding ourselves. You will simply have to write the genetic stock because once it gets registered with NBPGR, we add RG to the biological status. Then we have the elite lines. Elite lines is all the crop varieties that are under the varietal trials. So for that you can write EF. Then under, if the variety is released, it becomes an improved cultivar. It is a release variety ICRC. If it is a release, if it is a hybrid, it will be ICRH. If it is a state release, it will be ICSR. If it is an institute release, it will be ICIR. If it is an exotic one, it will be ICEX. But if you have a germplasm, if you have a germplasm or you don't know what is the biological status or you are doubtful about it, just simply write others. So that is how you have to fill in the passport data sheet. So this is all I have to tell. So thank you. If you have any queries, I would appreciate if you have any queries. I will be able to answer.
Participants are requested, please kindly interact if you have any doubts, queries. Hello.
Hello.
Hello. Your good name, sir? Hello.
Hello, Ravi.
Yes, ma'am.
In chat box also many questions are there.
Yes, ma'am.
Yeah, so madam can answer those questions. You can read
yes,
out. Okay.
Ma'am, Dr. Muhammad Raja, he asked actually how much quantity of seed will be taken in tetrazolium test. Hello. Anjali ma'am. Hello. 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 Anjali ma'am. Is it audible now?
Yes, you are audible.
Yes, ma'am. Okay, ma'am.
Madam.
Anjali ma'am, I think probably network issue, ma'am. Okay, meanwhile, we posted the guidelines and procedures of seed conservation at National Gene Bank, and please kindly refer that book. You will get a knowledge. It is a very informative book. That means what are the basic procedures and methods they mentioned by the speakers. Hello, ma'am, Anjali ma'am, is it audible? Yes, yes. Ma'am, Dr. Muhammad Raja, he asked how much quantity of seed will be taken in tetrazolium test, ma'am.
We will take 25 because the quantity of the seed with us is very low. We cannot just have a standard test like 200 seeds in here. But if you have to standardize for some crop, we can go for 200. But for gene bank purposes, we use only 25 seeds because we don't have many seeds in that.
And ma'am, Dr. Malarsve, she asked for how many seeds per accession tested for periodic monitoring.
Periodic monitoring, we do 25 seeds for application.
Yes, for periodic monitoring also.
But it also varies. We have to think because in some cases only we have 500 seeds in wild species. In that case, we cannot go for 100 seeds. So in that case, we will have to take 25 seeds for monitoring.
Yes, ma'am. And ma'am, what are the standards for sample size for submitting seeds for registration of germplasm?
It is 200 for 2000 for self and 4000 for cross pollinated, but for some difficult to seed species, it can be relaxed. For कोई ऐसा material जैसे MS line है, CMS line है, something अगर specialized material है in which it is not possible to have 2000 seeds, in that case is the relaxed also.
Yes, yes.
Some other question?
Regarding he is asking, ma'am, that is horticulture crops. What are the standards for sample size for submitting seeds for registration of germplasm of small seeded fruit crops?
Small seeded fruit crops में भी हम इतना ही लेते हैं.
Yes, ma'am.
Like जैसे Ziziphus all है वही conserve होता है. We don't conserve many of the fruit crops.
Yes.
In the seed gene bank, we don't conserve many of the seed crops. Maybe Ziziphus और ये जैसे बेर वे type का है, उसमें हम ले लेते हैं. 500 seeds we can take because it is not possible sometimes to have these much quantity. So in that case, we relax.
And when Dr. Malarsvi also asked how much quantity of stored per accession in MTS.
MTS, there is no standard for MTS actually, because in MTS we keep the material that transit or the material which has some problem or the material which has to go for regeneration. तो हमारे लिए तो MTS का वो meaning है. So MTS के लिए भी अगर आपको submit the MTS के लिए you don't submit us the seed for MTS. So MTS का कोई ये नहीं है. MTS में suppose you have collected the seed, you have collected only 500, we will keep in MTS. Then we will regenerate it, and when it is 2000 seeds, we will shift it to the long-term model. So MTS में as such कोई seed submission का ये नहीं है.
Yes, ma'am.
So if at your own place you are maintaining a MTS, you can keep large quantities of seed also, because from MTS you can distribute the seed. Active collection वहाँ पे रहता ना, तो you can even have half kg of the seed. It depends upon क्या आपको क्या करना है. What is your objective? If you want to conserve, then you can have 2000 to 5000 seeds. But if your mandate is to distribute the seeds, then you can have large quantities of the seed, multiplied seed in the MTS for distribution. That's it.
Yes. Any other participants? I hope everyone get guidelines, ma'am. Now it is the time for thanks to our speaker. Ma'am, Dr. Taraknath Saha sir also, he asked in PGR guidelines for getting IC number or registration of germplasm. There is no mention on number of cuttings or plants submitted to NAGs.
You can just see
Please.
25 to 50 लिखा हुआ है वहाँ पे. It is from 25 to 50. If he wants the guidelines, he could just send me a mail to me. I will just make the guidelines, highlighting what are the number of the accessions कितना करना. Normally हम 25 कहते हैं. We ask for 25 propagules जो भी है.
And ma'am, Dr. Malarsvi again, they asked for forest species collection seeds. How many trees to be collected and total number of seed quantity for storage?
Actually, forest trees में ये depend करता है, लेकिन क्योंकि कुछ trees ये तो ज़्यादातर recalcitrant या intermediate types पे होगा या ये field gene bank में ही conserve कर सकते हैं.
Yes, ma'am.
So यहाँ पर हम forest species mostly we don't conserve.
I hope. Okay, ma'am. Queries are completed. And if you have, please, if you have any doubts, you can mail or chat us also, and you can mail to directly to head of the division, Germplasm Conservation Division, and NBPGR. You can post. And ma'am, and thank you very much, ma'am. Thank you very much for your highly informative lecture and for guidelines, procedures, and methods. And we are all thankful to you, ma'am, for giving a nice lecture. And thank you, ma'am. Thank you. Thank you very much, ma'am. Yes, ma'am.
Thank you all the participants for listening, patience listening. Okay,
and dear participants, I am requesting actually one of our next speaker, Dr. Johnny Walt. He is busy with some business plan. Actually, that's why we are missing his lecture, and we are arranging his lecture at the end of session. That means at the end of our meanwhile 20 or 21 February, we are arranging his lecture. And now we can take some small break. That means we will meet at around 12:10, like that we will meet. This is unfortunately we missed his lecture actually, but even though we can arrange that lecture in last days. And please, we will meet and we will take some break, please. And sorry for taking this one lecture. We are missing, and we will meet at around 12:15. We will meet. Okay.
Rabi, 12, no? 12
Yes,
o'clock.
12 o'clock. Okay.
12 o'clock, yeah. So
12 o'clock.
The
Please
lecture
be
is
online.
very much important, very, very important. So
very,
we
very
also
important.
Be present there in that
Yes.
lecture.
It is a transitive conservation, and it is a new concept in conservation procedures. And don't miss that lecture. And she is Dr. Anuradha Agarwal. She is a vast experience in tissue culture and cryopreservation. And currently she is working as a coordinator in NEHP. At around 12 o'clock, we will gather. Please be online, and I will inform you in WhatsApp group before joining the speaker and other members. Please. Speaker. And I am requesting Dr. Jyoti, please kindly introduce the current speaker.
So whether
yes.
Slide is visible, Ravi?
No, not visible, ma'am. Ma'am, it is in half mode, not full presentation mode.
Okay, okay. Or I will. Sometime I will reshare again.
Yes, ma'am. You minimize the Zoom link app. Then make full mode, full presentation mode, ma'am.
Okay, okay.
All participants are requested kindly mute your microphone.
So is it no?
Ma'am, it is not in presentation mode, ma'am. It is.
It's okay, Jyoti, you can just.
Okay, ठीक है.
Yes, ma'am, you continue, please.
Maybe some issue with the.
Yes.
Okay, so today we have a very eminent speaker with us, Dr. Anuradha Agarwal, ma'am. So she is at present National Coordinator, CAST Program and Component 2 of NAHEP, that is National Agricultural Higher Education Project at ICR, New Delhi. Madam has acquired her PhD degree from Delhi University, and she has a long experience, more than 32 years of experience in plant tissue culture and cryopreservation research at NBPGR. Madam has the great experience of leadership also, as she has led the division of Unit of Plant Tissue Culture and Cryopreservation since 2017 to 22, where more than 14,000 accessions of orthodox difficult to conserve species, more than 800, those are conserved in the tissue culture or cryopreservation. Madam has developed several protocols for in vitro conservation and cryopreservation for medium and long-term conservation of very important species, that is Musa genetic resources. So she has developed very useful and cost-effective protocol for tissue culture and cryopreservation. So she has the pioneer research in cryobanking of Musa germplasm in India, and which included several crop wild relatives also. She has led several projects in the capacity of PI as well as co-PI. Around 18 national and international research projects she has handled. Then she is very dynamic. So she has served in many scientific committees, and she has provided her expertise, technical inputs on various aspects of science, agriculture, and policy. Furthermore, she has been a very good teacher. We can say the best teacher and very popular among the students. So she has guided 7 PhD and 13 MSc students from IRI and other universities. Furthermore, Madam has been very good organizer for all the programs. Still, she is in touch with our NVPGR, and she has been guiding us enormously for carrying out any events. And so she has successfully conducted 23 international and national training programs when she was at NBPGR. And now coming to the awards and honors. So the great prestigious award of ICAR, Punjab Rao Deshmukh Outstanding Women Scientist Award. It was bestowed upon her during 2011. So salute to ma'am that madam has achieved this award. So it is very proud for the NBPGR fraternity. And she has also received Boys Caste Fellowship long back, which is given by DST Government of India in 2001 itself. And then Madam is Fellow of Indian Society of Plant Genetic Resources, Fellow of Association for Improvement of Banana and Plantation. Then in addition to this, regarding the serving of the society, so our society, Indian Society of Plant Genetic Resources, so Madam is serving this society in various capacities since 2005. And she has organized successfully the first International Agrobiodiversity Congress in 2016, and second, that was in hybrid mode in 2021. She has, to her credit, more than 300 publications. So Madam will talk today, very nice lecture today about trans situ conservation of PGR. So certainly our participants will get the idea what is trans situ conservation, what is its importance. So ma'am, welcome you for this training program. So okay, thank you ma'am.
Thank you, Dr. Jyoti, for this very generous, I don't know whether I deserve such a wonderful introduction, but it is definitely, definitely, I am always happy to be associated with NBPGR and PGR activities. It is my bread and butter, even though I may have moved to another field right now. But thank you for giving this opportunity, and I will be very happy to share a little bit of experience that I have in the area of PGR conservation. So I'll start with sharing of my slides in a moment. Jyoti, you have to give me access to share the slides.
मैम आपको एडमिन को होस्ट बना दिया है, आप कर सकते हैं.
आप कर सकते हैं मैम. यस.
Okay. Okay, got it. Thank you, Vijay.
मैम थोड़ा प्रेजेंटेशन मोड नहीं आ रहा है.
अब आ गया?
नो मैम, डबल क्लिक करना चाहिए उसमें. हाँ, यस मैम.
ओके
ओके मैम.
Okay, is there a lag phase because मेरे यहाँ जल्दी आ गया था, आपके यहाँ थोड़ा देर आया?
2 या 3 सेकंड का गैप रहेगा मैम.
यस मैम.
अच्छा,
थोड़ा सा इतना लेके चलना.
Okay, okay. Thank you. Okay, so today we're going to talk about transitive conservation of plant genetic resources. And in the last few days, you must have got a very good introduction about what is the PGR conservation, management system that is in place in the country, how NBPGR is contributed towards it, and how other agencies are collaborating and doing this wonderful work in a partnership mode. And today, I suppose you would have had a presentation on conservation of plant genetic resources using the procedure of seed gene bank. Is that right, Jyoti?
Yeah, Dr. Anjali Kak ma'am, she has taken.
Yeah. So yeah, they have got an overview of how seed conservation is carried out, right?
Yes, ma'am.
Okay. So great. So I'm going to speak about how we are doing conservation in a more holistic and integrated manner, a concept that has been recently developed due to the experience we've had in the past. So typically, I think Dr. Anjali would have already apprised you about what are the broad areas or approaches that we have for conservation of plant genetic resources. Traditionally, like when we have started studying plant genetic resources, we were told that there are broadly two methods, in situ conservation and ex situ conservation. And as you can see in the slide, what are the various modes of conservation? So in situ comprises those methods of conservation where material, plant, crops, their wild relatives, weedy relatives, traditional varieties are allowed to grow in their natural habitats and allowed to evolve with the changing environment over a long period of time. So the conservation can happen in biosphere reserves, national parks, gene sanctuaries, and in case of agricultural crops, even within the milieu of the farmer's field, on-farm conservation. And I'm sure you'll have a detailed lecture later on this topic. So wherever we are allowing material to grow in its natural habitat, that all those conservation methods have been classified as in situ conservation traditionally. And similarly, ex situ conservation comprises those methods where we remove the material from its natural habitat and take it to another site, either in the field or even in the laboratory. So this is practiced mostly in cultivated species, wild relatives, land races, traditional varieties, etc. Now, already in the morning, you would have had a detailed presentation on how we carry out conservation in seed gene banks. And later during the course of your training, you will be apprised about use of tissue culture techniques for conservation through in vitro gene bank and cryopreservation techniques through the use of cryogene bank, and maybe even DNA conservation in the form of DNA bank. Whereas in the field, we can have field gene banks, arboretum, herbal garden, and botanical gardens. So I am sure all of you would have had some idea about these forms of conservation. And initially, when conservation programs started about 50 years ago, these categories were like kind of distinct from each other. So if you started a seed gene bank program, you normally did not have a herbal garden. But over a period of time, it was realized that if we have to do conservation, now conservation principles is what is important to know before we go for deciding a conservation program. Population biology of a particular species or an ecotype or genus is very important to be determined before we determine what kind of method has to be applied for its conservation. And while I think you must have had a lecture on exploration, how we sample germplasm in the nature, select the correct population to capture the maximum number of alleles, and put it in a particular method of conservation. So each of these conservation methods have certain guiding principles. And over the years, technical guidelines have been developed for their conservation in each of these methods of conservation. NBPGR also has made a publication, and I'm sure Dr. Jyoti would have shared one guideline, management of plant genetic resources. Dr. Jyoti, I hope you have shared with them.
Yes, ma'am. Some information we have shared, and some we will be sharing all the information related.
No, I'm talking particularly about the guidelines on management of PGR that was published in 2016.
Yes,
that we have not said, but we will be sharing, ma'am.
Yeah, please share those.
Okay, yes, ma'am.
It will be very useful for all the people in this group to know how principles, basic principles, do's and don'ts need to be followed for managing each material in each of these facilities, right?
Yes,
so very clear-cut guidelines have been developed, how to do sampling, how to do processing, how to store the material, how to regenerate the material, and how to use the material. And these days, of course, everybody talks about sustainable utilization of conserved material. So those guidelines will give you a broad overview of how germplasm conservation is carried out at national level, at local level. At international level, also there are guidelines by the FAO, which again our organizers can share with you. Guidelines for seed conservation, in vitro conservation, cryoconservation, field gene banks, and of course, in situ conservation. So that literature already exists, and these are the basic starting points for conservation. Because time is limited, I will not go into the principles. I will just talk specifically about the topic of trans situ conservation, which is a relatively a newer concept. Okay. While I'm speaking, you are very free to stop me in between and ask any question. Okay. Now I'll introduce you to a few more new words in this arena of conservation. So by now, I hope it is clear to you that traditionally in situ and ex situ conservation methods were considered to be exclusive to each other. So wherever you're doing ex situ conservation, those people were normally not associated with in situ conservation. But in the recent past, with the greater rate of extinction of species, loss of land due to many reasons, climate change, etc., etc., it has been increasingly realized that we need to have an integrated approach of conservation. That means we apply more than one method of conservation, and this can vary. This can be within the in situ methods, within the ex situ methods, and more recently, it is realized it's important to have both in situ as well as ex situ methods together to conserve either a crop or a population or a whole set of genera or species. So a large number of these integrated methods have been defined in the last, I would say, 1 decade or so. And I'm just giving you an introduction to those terms. So the first is this in situ method of conservation. That means a combination of in situ methods. Okay. So this is offsite collection maintained within a natural habitat and is in a sense in situ reintroduction strategy. This is mostly being applied to species which are under the threatened category. I think all of us would be familiar with plants falling under the threatened category. That means the number of populations in nature has reduced drastically. There is a genetic drift or a genetic shift, and these need higher priority for conservation. So in these methods, mostly species are threatened in nature because of habitat loss. You know, we cut down forests, we change the use of land, like from a natural population, we convert it into agricultural land or into construction sites. And then there is a rapid erosion of species. Now, this method, what it does is it introduces those species which are threatened in a similar ecological zone as per the original species. So in the natural habitat in which the species existed, if one site is eroded, species are introduced close to that site so that when the species gets established, it has a natural ecosystem in which it was adapted, and it slowly over a period of time, the natural ecosystem resembling the original one gets established. So it can be applied to the lands of low economic value, such as wastelands or abandoned agricultural lands. And it allows simultaneous reintroduction of a large number of species. So this method has been applied in a few ecosystems, both in India and abroad. Then coming to the next method is the forest gene banks. Now, forests, as you know, are the best places in which species can be conserved, because a whole range of species get conserved. And in fact, not only plant species, but animal species, soil microflora, everything gets conserved easily. But with the rapid loss of forests themselves, many of the species are getting eroded. And this particular species has a method of taking a natural forest. The forest gene bank itself is the in situ site, where a particular existing population acts as an in situ sink, into which genetic material from several other outside sites is introduced and maintained. So for instance, if there is, say, a sal forest of one provenance in one district, the forest gene bank concept uses the in situ forest to introduce material from other provenances, but for the same species. So this is a combination called as forest gene banks. It's not forest per se, and it is not a gene bank. It is a hybrid between the two. So forest gene banks have been conceived, in fact, in some of the Indian species also, and being followed for maintenance of material, large number of diverse species. Then the next, so this particular slide gives you the how forest gene banks are established. Again, there is a systematic do's and don'ts about mapping the geographic species, distribution, and identification of the ex situ donor plants, and the process of gene introduction from source to sinks, because once you're introducing material from another provenance, there will be outcrossing and a change in the genetic structure of the area. So a lot of scientific information needs to be determined before we can establish these forest gene banks. Now, the next hybrid method of conservation is the Circa Sutu method, which is a farmer-based conservation method that has been applied in mostly horticultural species. In this, the conservation is done for plant species or remnant trees in farmland or altered agricultural landscapes, such as agroforestry systems or home gardens that are outside the natural habitats, but within the native geographical range of the species. So if there is a tropical ecosystem in which the species was native, you do it not in the same geographical area, but in the same geographical ecosystem. So this conservation method has been applied and it integrates conservation with the socio-economic trends of that area. Whereas in the in situ and ex situ type of conservation, it aims to remove the areas and species from the influence of those trends. So here we allow the socio-economic trends to occur. And this Circa situ method of conservation, it helps in maintaining the biotic and abiotic interactions and allows populations to continue to evaluate the process and generate genetic variability. See, ultimately what we are doing in each conservation program is to either ensure that we maintain the genetic gene pool as is available, and in others, we try to maintain genotypes. So whenever we are doing these integrated conservation methods, we have to make sure that all these genetic interactions are scientifically documented and help in the ultimate use of the material. So this is just the depiction of the circa situ conservation. So it is an integration of the natural habitat of agroforestry and home gardens in the same native geographical range. Then there is also another integrated method called plant micro reserves, not very common in India, but it is being practiced outside. The plant micro reserves are small sites that are protected by law to preserve unique or threatened flora, to manage the habitat according to the conservation needs of those species, and develop conservation experiences guided by the technical and scientific research. It basically involves preserving species genetic diversity through a network of small lands, about 2 to 20 hectares, which encompasses a large number of populations in different natural habitats. So here the focus is on habitat preservation along with species conservation. And normally this occurs in highly fragmented landscape. But since it is linked with a legal provision, it is not applicable in many of the countries, depending on the landscape. So plant micro reserves are not still very popular, but being practiced in smaller areas with diverse fragmented lands, as given here in Western Crete example. You can look up the paper. Then more popular is the quasi in situ conservation. Here it comprises using ex situ collections in natural or semi-natural environments as a part of a complex ex situ in situ conservation strategy, preserving both neutral and adaptive genetic diversity. This involves creation of depositories of genetically variable source material for species of conservation concern as living plants maintained under natural or close to natural conditions. So the difference over here is we allow a little bit of natural breeding and evolution to occur, whereas in an ex situ milieu, we do not allow that evolution to occur. So this is a transition between ex situ and in situ conditions. So that's why the word quasi in situ condition. So again, there are standard steps to follow how to establish a quasi in situ model. It involves survey analysis of species distribution, sampling of populations according to a spatially or ecologically structured design, planting in appropriate sites with natural or semi-natural habitats close to the environmental match, and for maintaining the collections. Very importantly, studying the life history, traits, and biotic and abiotic effects of the population demography, and reintroduction or reallocation, preferably using seeds from the living collection and monitoring of relocation success. And this model is specifically used for species where the seed output is low and it varies greatly between years, or reproducing adults in natural populations are very few and far apart. So here, quasi in situ conservation. So most of the threatened species are now being conserved using this quasi in situ model. And there are many examples in India where this is being practiced, where threatened material is removed from its original site and brought to this site where it is initially controlled and then allowed natural population to develop. Now, so this is the model for the quasi in situ model, and you can read this paper by Wallace et al. To get greater details of how this is carried out. Now, Coming particularly to crop genetic resources, which is the focus for the PGR management system that we work in. Over the years, it was realized that we need to have a combination of these strategies, especially in situ and ex situ strategies, to be combined to cover the entire range of species that occur in nature for a particular given crop or crop group. So this term trans situ conservation is particularly therefore now applied for the whole range of conservation strategies adopted to conserve the entire genetic diversity of a given crop or crop group. And I will illustrate this, how we are doing it in our system using 3 examples of banana, taro, and allium species. So this is a systematic approach of integration of multiple in situ and ex situ approaches. I have already explained what are the in situ and ex situ approaches. And the most important feature over here is we provide in situ conservation for management and research. Also, we'll take care of local and regional seed and living plant collections for conservation, research, and education. And the conservation also involves national and global gene banks maintained for plant breeding and crop research and exchange of germplasm. So to cover the entire gamut of both conservation, use, and exchange is what we call as the trans situ conservation of PGR. So it's basically a very, very scientific amalgamation of in situ and ex situ conservation to achieve capture an entire gene pool of material. And this is especially useful in horticultural material, where unlike some of the traditional cereal crops or legume crops, where a large number of material can be conserved in the form of seeds, horticultural material or clonally propagated material requires a trans situ approach. And I will illustrate to you how we have been doing it. So as you would have already come to know how the PGR system exists in the country, we have the gene bank, the National Gene Bank at NBPGR, comprising seed gene bank, in vitro gene bank, cryo gene bank, and field gene banks. So all the ex situ methods for conservation, which is where NBPGR is one of the agencies, rather the nodal agencies to carry out these ex situ conservation of methods. But collaboration and networking with state agriculture universities, ICR institutes, other central universities, farming communities, and other stakeholders, even tribals and forest dwellers, is what has been adopted in the recent decade and a half to carry out a more holistic conservation. So going across ex situ and in situ, involving a large number of stakeholders and carrying it out in a complementary, mutually beneficial manner. So I'll start with the example of banana. As already Dr. Jyoti mentioned, I have worked a great deal in banana. And I think all of you would be familiar with the immense genetic diversity of banana in the country. So right from very primitive diploid species which are seeded, especially the Musa acuminata and Balbisiana, down to the very advanced cultivars which are growing on a commercial scale, from diploids to triploids, and we even have tetraploids. A large genetic diversity of Musa exists in our country. So about 31 species to 39 taxa of banana have been documented, belonging to various sections, a large number of which are endemic. And the wild species especially are distributed mainly in the Eastern Ghats, Western Ghats, Northeastern India, and in Andaman and Nicobar archipelago. That means all the biodiversity hotspots in the country carry a large number of diversity of this species, this particular genus. And in fact, the Musaceae family to which banana belongs has only two genus, Musa and Ensete. So we also have a large number of Ensete diversity in the country. And the cultivated diversity, the bananas that we eat, which are mostly triploid, there's a huge diversity in the country. Already 400 to 500 accessions have been documented, collected, uh, evaluated, and bred, and also being conserved, which belong to the various genomic groups coming derived from Musa acuminata and bulbiciana, the AA and BB genomes, and a huge number of subgroups. So although banana appears to be one crop, in terms of diversity, it is an immensely diverse species. I mean, it's just amazing to see how much diversity exists in just one crop. And this diversity derived from different modes of reproduction. And therefore, this particular genus required a multi-pronged approach for its conservation. Now, like I said, most of the bananas are triploids. So therefore, they are sterile because they are parthenocopic, and most of the reproduction is carried out vegetatively through the means of suckers. Whereas the diploid species in which seeded bananas exist occur especially in the forested areas, and they can be produced both through seeds or through vegetative means. And therefore, we realized initially what we considering it is as a vegetatively propagated crop. There was only the approach of conserving it through field gene banks as well as in vitro gene banks. But once we realized the whole reproductive biology of the plant, it was understood that both in situ and ex situ approaches were required for conservation of the entire gene pool. So over the years, we have developed methods for conservation in nature reserves, in on-farm conservation, in cryogene banks, in seed gene banks, in DNA banks, in vitro gene banks, and in field gene banks. So, and each one of them supports the other conservation method in a very complementary manner. And this we have realized after studying a detailed reproductive biology of the plant, and how some of them we can just simply conserve the seeds, both in the seed gene bank as well as in the cryogene bank. Whereas others where no seed production was available, we have been putting them in tissue culture gene bank or in the field gene banks. Besides, some of the sites, especially in the Northeast, have been marked to conserve the plants within the nature, especially in the Northeast as well as in the Western Ghats, to allow the wild species to evolve and perpetuate in nature itself. So to begin with, I don't know how many of you are familiar with the fact that tissue culture of banana was thought to be an impossible task. It was considered to be a recalcitrant species, but with research and development, tissue culture is now a very well established industry in bananas. Therefore, we could establish in vitro gene banks followed by cryogene banks. And I'll just briefly show you how. Today, I am not using this opportunity to show you any method, but to tell you that when we have to capture the diversity in a crop, all these methods become very important. And I'm just illustrating it here. So based on these conservation approaches, we need different types of material that can be conserved, right from whole plants down to the DNA, including its pollen, tissue culture, explants, plantlets, and seeds. So this whole entire gamut of diversity is captured. And since the last 3 decades, this was being done through a collaborative mode between collaboration between ICR institutes, primarily in National Research Center for Banana, in collaboration with NBPGR, many state agricultural universities, in fact, the ones that have been having the centers for All India Coordinated Research Projects, and a few state, central and private universities and institutes in the country. So what I'm trying to tell you is trans-situ conservation involves a network, and this network is very important for developing the conservation program and sustaining it in the long run. So over the years, we have been supported through various projects also, right from ICR to DBT to DST, World Bank, Biodiversity International, Global Crop Diversity Trust, and many international fundings like coming through IITA, etc. So through these networks and externally funded projects, plant exploration started early during the 1980s. We had a lot of initial collections done through our regional station Thrissur, and where they established a field gene bank. Thereafter, the field gene bank was shifted to ICR NRC Banana at Trichy. And during the NATP project, which was conducted during 1999 to 2005, intensive collections were made under this project, and a large diversity was collected. And a very systematic field gene bank could be established at NRC Banana and Trichy. It is one of the Asia's largest Musa and Ensete collection, comprising more than 480 accessions. Both indigenous and exotic, because as you know, the world over, a large number of different Musa species and genotypes exist. There is a world collection of banana at Leuven in Belgium, and we have been collaborating with them also. And over the decades, a systematic collection and evaluation of in the field gene bank, both in Trichy, and they have a satellite gene bank at Agali, have been established, material has been evaluated, and a whole network of germplasm is distributed between the egg crib on fruits. Some of you, of course, must be familiar with this, or maybe working in one of the centers, if not for banana, for other egg cribs. And this collection has been characterized, evaluated, used for breeding programs. A lot of varieties have been developed. A few of them have been released and registered. And to overcome the problems of field gene bank, we have established the tissue culture gene bank, both at NBPGR and the National Research Center for Banana. And to overcome the limitations of tissue culture, because you'll get the details of these techniques in subsequent lectures from Dr. Sandhya and Dr. Malik. So I'm not going into those details of how the technique is applied. We have subsequently established the cryogene bank collections, both using large cryotanks with liquid nitrogen in vapor phase as well as in the liquid phase. So this particular flow chart shows you that carrying out collections, both for ex situ especially, requires a detailed SOP where there is clean material which requires to be cleaned. Only disease-free material is conserved in tissue culture or cryogene bank. There's a whole science of applying genetic stability testing, because when we are doing tissue culture, soma clonal variation becomes an issue, and how data is maintained in the gene database. And tissue culture technologies require hardening and field transfer technologies. Those are also standardized. And ultimately, whatever conservation we do is used for distribution to partner institutes, as well as in India as well as abroad. And that is done through the methods of exchange that have been told you, using MTAs, etc., etc. Now, so over the years, using these in vitro and cryotechnologies, different genomic groups have been conserved, which have been also brought out after conservation for a long period of time. And they have been used for developing cryopreservation protocols. Again, you will be getting the details in the subsequent lecture, but developing cryopreservation protocol is a very high-end research, and we are very proud and happy to have a great expertise at NBPGR, especially in the tissue culture and cryopreservation unit. You will get more details about it later, but I can answer any questions. So protocols have been defined and used for conservation of material. And like I said, we also have seeded bananas, a huge variety of wild relatives and wild species, where we have used the seed to conserve both using seed gene bank and cryo gene bank, and we have found it works equally well. So now we have a standard protocol using which we can use any seeded banana for conserving under in vitro conditions and cryopreservation conditions. Now, while doing research, people have also developed embryogenic cell suspensions. Again, these can be conserved in the cryogene bank, and that is another complementary aspect of conservation. And NRC Banana has also developed a DNA bank, where DNA is extracted from the young leaf and kept for molecular studies with the related data available in the data bank. Again, transgenics is something that is adding to the gene pool of a natural gene pool of a species. Although man-made, there is genetic diversity being created. And again, in banana, a good success has been achieved for developing transgenic lines. So these are maintained under controlled net house conditions. So like I said, based on the need, on the biology, one or the other conservation method can be applied for conservation of the material. Rather, it needs to be applied. Now, I had mentioned something about on-farm conservation. So here what we do is we involve the farmers, the tribal communities to participate in conservation of material. So here what NRC Banana has done is to develop disease-free planting material of a particular niche. So this is in Koli Hills in Tamil Nadu. Two extinct varieties were multiplied in tissue culture, provided to the farmers, and they have now re-established it in the same area where the plant was, where these particular varieties were getting extinct. So this is again adding to the complementarity of the material. And all the material that we have been collecting, conserving is utilized by sharing within the country and outside the country to the users. So conservation with use is a very important aspect of this trans situ conservation method that we are talking about. So over the years, a lot of material has been shared and utilized wonderfully for the country. And this is how we have been working for bananas in the NBPGR along with its partners, and we continue to enhance the program in future. Now, similar to that, the same principle has been applied in many other crops, but I'm just taking two more examples. One is garlic, which belongs to the genus Allium, and it's one of the largest monocot genera with more than 1100 taxa that are present the world over. And in India, we have a very rich diversity of garlic. And since 1986, concerted effort again has been made towards collection, conservation of both cultivated and wild alliums. So in alliums, of course, allium cepa, which is onion, they produce seeds and they can be easily conserved in the seed gene bank. A large number of species have been conserved because they produce the orthodox seeds. But many of the allium garlic species, Allium sativum and the other wild species, do not produce seeds, and they have been conserved using both in vitro gene bank and cryo gene bank, as also a large number is maintained in the field gene banks. So again, a complementary method to capture the entire gene pool. Cryogene banks offer long-term safety, whereas field gene banks give the ready-to-use material for breeding and exchange. The in vitro gene banks are a safety net in between. Wherever we are not able to do cryogene banking because of lack of protocol, the in vitro gene banks are useful. And like I said, seed gene bank is only applicable in species where seeds are produced in nature. So garlic and its related species are ones that either do not produce seeds. Garlic does not produce seed. Related wild species produce seeds. So some of them are put in the seed gene bank, but most of them are in the field gene bank. And now we are making efforts to put the vegetative material in the cryogene bank. So based on the biology again, whether we want to conserve gene pool or we want to conserve a genotype, the conservation strategy is applied. And you can see some of the examples of the various alien species that have been conserved in tissue culture at the NBPGR in vitro gene bank. Around 160 accessions of 10 different alien species are being conserved under tissue culture conditions, both at 25 degrees and at low temperature. So again, details will be provided in the tissue culture lecture subsequently. But just to give you an overview how various protocols have been developed to conserve the material, diverse protocols to conserve the diverse materials. So we have low temperature conservation of cultures. We also have production of, these bulblets in the tissue culture itself, which can be conserved on tissue culture medium, or they can be just harvested and kept in cryovials and conserved for 2-3 years, and they do not lose their viability. So a large range of strategies have been developed to capture the entire genetic diversity of the different species. Again, a huge number of cryopreservation protocols have been developed. I'm not going into the details, starting from shoot tip explants, how we cryoprotect the material to allow it to tolerate the very extreme low temperatures of liquid nitrogen. Similarly, we have been applying the same trans situ approach to conservation in another crop called taro. Taro is a tropical eraceae plant, monocot. It's widely grown in the country in various states. And due to monoculture, a lot of the traditional land races have been lost in the recent past. So again, we have done this exercise over a period of 3 decades to collect material, bring it in the NBPGR to bring it under field gene bank, which is being done at our Thrissur station in Kerala. And the in vitro material of the same accessions has been brought under in vitro gene bank in Delhi. And often after a few cycles of conservation, we regenerate the material back in the field. And very recently, we have shared some of the old traditional varieties that were collected about 2-3 decades ago. And at the request of farmers, we have reintroduced it in the farmers' field. So what has happened over the years, many traditional varieties that were lost in the field could be repatriated to farmers' field because they were conserved in our gene bank. And this is one of the farmers, Mr. Regi Joseph, who asked for one of the taro materials, in fact, a large number of taro material, and we could provide him the material. This is from Regi's team. And this is Dr. Vartika, who is now doing both banana and taro work. She's also co-author in this paper. So this material is under field evaluation with Mr. Regi, and he is sharing it with the other farmers in his countryside. So what I'm trying to tell you is conservation has to be linked with utilization, re-establishment, reintroduction to be successful in the long run. Otherwise, if we just keep it in the gene banks, it becomes, static material that neither is being used nor is being shared. So again, a large number of accessions of taro, more than 400, are under conservation in field gene bank. So not only our regional station in Thrissur, but also the CTCRI at Thiruvananthapuram. In fact, they are having the largest collection in the field gene bank. We work in collaboration with them to complementally get material for in vitro conservation as well as for crab preservation. Crab preservation is still at its early stages. There are a lot of problems associated with plants like taro, like endogenous bacteria, and we need to, so research has to continuously be integrated into developing these trans situ methods of conservation, integrating one method with the other, deciding which crop group requires what kind of conservation. That constantly requires research to take place. So these are my last slides. It just pictorially captures how we are doing trans situ conservation. In case of banana, we have both in situ and ex situ methods, which I have already explained, and how they are intermittently being exchanged between the techniques for constant use and increase in diversity. Similarly, for allium genetic resources, in situ and ex situ methods are complementing each other. So our field gene bank in allium is at Bhowali. The in vitro gene bank in all the cases is at Delhi, as also the cryo gene bank. And similarly in the case of taro. So this was just to give you an overview of how multiple conservation needs to be carried out simultaneously to ensure that we are not losing any genetic diversity, and also facilitating expansion of that genetic diversity while we are doing the conservation. So truly a dynamic conservation system where the most important keyword is sustainable use. People get to use the material and we do not lose the material, to put it very simply. So these have been the efforts in our trans situ conservation. We perpetuated, especially for horticultural genetic resources, where conservation becomes tricky if we only adopt one method. And the only difference, I don't know whether in the morning you were told about redundancy in gene bank, duplication of collections. Here, in fact, in seed gene bank, while we say we try to avoid duplication of accessions, in trans situ approach, redundancy is encouraged, because if there is any loss of material in one of the methods, and there are live examples with us now. For instance, the Icada gene bank in Syria had to be totally relocated because of the war. So war, climate change, natural disasters make us realize that safety duplication and having a collection in more than one places is important in the world, in the very volatile world that we live in today, where things change, things happen which we had not foreseen. Because at the end of the day, genetic diversity is very important for our agriculture and therefore food security. So this is the model that we have adopted. And now the topic is open for discussion. Before that, I would like to thank the organizers of the training program, especially director and staff of NBPGR, all the funding agencies, all our network partners over the years, and all especially the staff of Tissue Culture and Crop Preservation Unit, where I have worked for almost 3 decades, for giving me this opportunity today, and for of course, the work that I have done over there. Thank you. So over to you, Jyoti.
Okay, thank you, ma'am. So now I will request to participants, if they have any question, they can ask. Ma'am, the session is open for discussion, or they can put in the chat box. I will read out from there.
So I understand that I did not give details of the technique because there are subsequent lectures in that, but it's more to give a concept of those who will be getting associated with our conservation network, what is our approach and what is our expectation. But I can answer any technical questions for techniques also, if anybody wants.
Okay, it was very informative.
Or it went above everybody's head.
I'm not seeing any question in chat box. I have shared, ma'am, the guidelines, the link I have shared for the conservation aspect, and we'll be giving the link for all the relevant literatures available on our website. So those we'll be sharing to all the participants.
Okay, I see one question. How to maintain inside and outside temperature of transgenic net house? Okay, yes, so if it is a net house, if it is a net house, Dr. Mohammed, where are you? In Kazri?
Yes, ma'am, from Kazri is there.
In Leh. Okay, that's why his question. Yeah, that's a good question for a place like Leh. So the transgenic net house that I have shown you for NRC banana, since they are growing it, they have established the net house in the native place where the plants grow. The temperature control is not required. But if you are in a place like Leh, and depending on the biology of the crop, then there are very standard greenhouses, controlled temperature greenhouses that are available, from very small to very large ones, where temperature can be artificially maintained with not very high cost. So you are asking because you're located in Leh. So wherever, if you are doing this kind of work in Leh, naturally you will need to have controlled conditions, unless you are doing native plant establishment, plants belonging to Leh region, then just a net house will work. Otherwise, you need a poly house or a glass house. Anybody else?
So we'll be sharing the contact of all the resource persons. So madam contact detail will also be there in the, so later on also means they can ask if they have some query.
Yeah. I understand this was very generic for them, because once they had done the techniques, then my lecture would have been more understandable to them. But nonetheless, I just wanted to put the concept across.
Yeah, it was very new, novel concept, trans situ conservation. So I also enjoyed very much listening all the things and got acquainted with every details. So now I'd like to, Dr. R.K. Gautam, sir, want to thank you, ma'am. So I'd like to invite Dr. R.K. Gautam, sir. Sir, please.
Okay. So, namaskar, welcome, madam, once again to our training program. And I think it was a befitting treat to all the trainees to listen to Dr. Anuradha Agrawal, who is our own NBPGR family member. We appreciate her very long and outstanding experience and expertise on in situ mediated PGR conservation and management. Besides scientific excellence in application of cryopreservation for PGR management, she has excellent management, coordination, and administrative skills. So that is your another great strength, madam. We appreciate your experience and expertise, and we are grateful to you for having accepted our invitation to share your excellent work and expertise with the trainees and with all of us. I was attending the lecture throughout its course. On behalf of organizing committee, I express our sincere thanks to you. We look forward to your continued support in our all the academic endeavors in future as well. Thank you very much, ma'am. Thank you.
Thank you, Dr. Gautam. The pleasure was all mine, and happy training for the rest of the days.
Thank you so much, ma'am. Thank you. Thank you.
Thank you. Bye.
Thank you, ma'am, for your good wishes.
Yeah, and thank you, Jyoti. And I thank all my co-authors, especially Dr. Gautami, who has really helped me in putting together this information, as well as Dr. Vartika and Dr. Subhash Chandra.
Okay, ma'am.
Thanks a lot.
Thank you, ma'am.
Thank you, ma'am.
Thank you. Bye.
Okay, so next session will be from 2:30 onwards, and Dr. Ravi will take up the next session. So everybody should join at 2:25 p.m. Thank you, one and all.
Procedures for long-term conservation and of orthodox seeds by Dr. Chitra Devi Pandey and Padmavati Gore. And let me introduce our current speakers. And Dr. Chitra Devi Pandey, she is a principal scientist in the division of germplasm conservation in NBPGR, and she is a curator of vegetable genetic resources at Indian National Gene Bank, and she involved in the trait-specific information of vegetable germplasm of various sources, and that means rationalization and documentation of one. And next, she is a main contributor in development and standardization of breaking seed dormancy in wild solanum, abelmuscus, Thespecia, momodica, and Corcorus species. And she developed a quick viability test in Jatropha and Baliospermum. And she undertaken the seed storage behavior. And she worked out in Aegil marmalus, Rheum emodi, and Rheum murcrophytianum, and Rheum astral, and Wrightia tinctori. And she development of best practices of executive conservation of radish germplasm for crop gene bank knowledge database. And at the same time, she involved in the development of global strategies for conservation of genetic resources of cucurbits, brinjal, and sorghum in collaboration with Crop Trust, Germany. And she registered 12 unique germplasm materials, out of which 6 are vegetable genetic resources and 5 are millets group. And she is having a patent or copyright one, and publications are more than 55 referred articles and other publications, which I mentioned in this slide. And she is a curator of vegetable genetic resources and extensive work developed in vegetable genetic resources, and mostly brinjal, cucurbitaceae members and others. And she is a fine expert in the field of this long-term conservation of vegetable genetic resources. And next participant is, and she is Dr. Padmavati G. Gore. She is a scientist in division of germplasm conservation. And she is a curator of legume genetic resources at Indian National Gene Bank at ICR NBPGR, New Delhi. And she developed a dormancy breaking protocol in wild Vigna species. And she delineated in taxonomic keys of wild Vigna species. She registered in 11 unique germplasm materials. And she is a best outgoing student from IRI. And she received recipient of KL Mehra Best Memorial Best PGR student. And as a reviewer in several international journals, and she published more than 50 research papers and other publications also there. And madam, and I am inviting you, both of you. It is my great honor, and please kindly share your experience and knowledge or expertise in the conservation of and long-term procedures of how methodology and methods are in vegetable genetic resources. And please, ma'am, please welcome to both of you, and please continue, ma'am. Yes, ma'am.
Thank you very much, Dr. Ravi, and good afternoon to all the trainee participants. I am Dr. Chitra Pandey, as he introduced, and I am involved in conservation of vegetable crops mainly. And Dr. Padmavati Gore, she is handling leguminous vegetable germplasm. Coming to the topic which allotted for the practical is processing of orthodox seeds for long-term conservation in the National Gene Bank.
So, sir, our slides are visible?
Not
is
yet,
it,
actually.
It
it is one empty screen is coming, actually.
Your screen sharing is paused.
सर शेयर किया है हमने.
अच्छा, no, you move this second slide. Actually, it is some blue screen is coming. Okay. Yes. But it is not in presentation mode. थोड़ा आप एक बार अनस्क्रीन करके फिर पूरा ओपन करके शेयर स्क्रीन करके.
ठीक है सर.
नो थोड़ा छोटा ही है एक्चुअली.
फिर चलो उधर चलो.
ठीक है वो. ओके ओके ना.
हाँ मैम, ये तो
ओके
पूरा
ओके
स्क्रीन नहीं आ रहा है मैम. हाँ पूरा स्क्रीन नहीं.
ओके
presentation slide which is in presentation mode. At that time you just share it.
No, we are keep here it is on presentation mode only.
Okay.
Okay, let me share it from other window.
You minimize the Zoom link, ma'am. Then you open the PowerPoint.
Yeah, we open.
Yes. Sometime, ma'am, it will come late actually. No, नहीं आ रहे एक्चुअली.
सर अब देखिए.
No, अभी भी नहीं आ रहे पूरा.
ओके फिर वहीं से करो. चलो बताओ.
सर प्रेजेंटेशन मोड पे है अभी.
नो नहीं है. नहीं तो आप एक बार अनस्क्रीन करके फिर डबल क्लिक करिए वो स्लाइड में, फिर पूरा आ जाएगा.
You please move this window away from the shared application.
पेन ड्राइव ले लो.
Ma'am, meanwhile, for your information, ma'am, actually, the participants are from 18 organizations, and they are near around 84 participants are there. Some are not able to attend due to the network issues, but mostly they are attending.
Good number of participants from 18 different institutes.
Yes, ma'am.
सर आ रहा है अभी.
हाँ अभी पूरा आ रहा है. यस. हाँ
ओके. एक मिनट सर.
हाँ यस
मैम अभी आ रहा है, इधर ही आ जाइए चित्रा मैम.
Okay. Yes, it is visible. Yes ma'am, continue ma'am. Yes.
So आ रहा है.
आ रहा है मैम, वॉइस भी अच्छा आ रहा है.
सब कुछ आ रहा है.
Good afternoon to all the participants. Coming to the allotted practical, that is processing of orthodox seeds for long-term conservation in the National Gene Bank. Today morning, Dr. Anjalika, she explained about the different activities which are carried out in the National Gene Bank. Coming to the main objective, that is what is the aim of conservation? To preserve the original genetic characteristics and viability of an extraction for our posterity purpose, for which optimum seed storage conditions are needed to ensure a prolonged seed storage life. So we have to keep prolonging our seed storage life so that we can able to supply viable and useful germplasm to the users. Users may be anybody, farmers, breeders, researchers, students, or anybody. So we have to keep good viability in the National Gene Bank. So all of you know about the types of seed storage. There are 3 different types of seed storage behavior. One is orthodox seeds, which can withstand dehydration or desiccations. Means you have to reduce the moisture content before conserving in the National Gene Bank. And orthodox seeds are said to be like tolerate to low temperature and low desiccations. Whereas intermediate seeds, it can withstand dehydration to a certain extent. That is up to 10% moisture content you can reduce, after which the seed viability will be lost. So you cannot store such type of seed. For example, neem, citrus, all those things. So you have to go for the alternative methods such as field gene bank or cryogene banks. Coming to the recalcitrant seeds, the seeds which cannot withstand dehydrations, which can tolerate low temperature. So some of the examples are mango, jackfruit, and horticultural crops which requires more moisture to survive for longer period. So alternative method is recalcitrant, that will be dealt by cryopreservation unit. So what we are receiving, more than 85% of the seeds in the national gene banks are said to be orthodox seeds. So presently we are conserving more than 4,63,000 in our national gene bank, which consists of more than 10 crop groups. So what are all the advantages of long-term seed storage? And it is the safest and the most convenient method. The main advantage is cost-effective and it requires less area, ease of accessibility and documentation, and all the samples what we are receiving are subject to plant quarantine system. So in the local system, so those are all free from pest diseases and other hazards. So we are conserving all the germplasm which are subjected to quarantine. The main disadvantage is we are freezing the evolutionary process because these are all man-made structure and we are conserving in the National Gene Bank. We are not allowing for the evolutionary process to occur in the in situ, how it is happening. And the total genetic integrity cannot be assured because the samples which we are collected from different areas may have contaminations, especially in cross-pollinated crops. And sampling size is also again difficult to process. The wild, weedy plant, endangered species, and other cross-pollinated crop, it is difficult to ensure the adequate sampling. Coming to germplasm conservation facilities, I think this you might have seen in so many presentations. Just I would like to emphasize, this is our national gene bank, which is the upgraded one. Orthodox seeds can be stored over here, and we have got 12 different modules, which are all maintaining at -18 to 20 degrees centigrade. And these 12 modules are allotted for different crop groups. So crop curators are responsible for all different modules. I am handling vegetable crops, and my allotted module is 9, and we have all security system, so I can access my module only. Similarly, like vegetatively propagated material, we have got in vitro gene bank and field gene bank, and the recalcitrant type, endangered, wild and weed species cryo bank is again used. The important point what I want to emphasize is precautions what to be taken before sending the germplasm for long-term conservation. So one should know, like well-developed and physiologically matured seeds can be sent to gene bank, and with most care should be taken to avoid harvesting in rainy season. Seeds should be dried soon after harvest, and it should be shade dried, and the moisture content should be around 10 to 12%. Sheets should not be treated or coated with any chemicals. Most of the times in case of released varieties, we are receiving with coated or treated with different chemicals from private companies and other government organization also. So such type of seeds we will directly reject it. We won't process it. And it should be free from pests and diseases and free from weed seeds and other crop seeds. It should be of pure cultivar or germplasm what you have collected, which have to be stored. And it should be clean and free from undersized, shriveled, immature, and discolored seeds. All the germplasm or seed, whatever you are sending, it should be properly labeled, whether it is the collector number or any identity what you are writing, it should be properly labeled and packed to avoid damage during the transit. If you are sending in the muslin cloth bag, you have to put at least one handwritten label inside that. On the top of it, like in the muslin cloth also, you can write the name. Similarly, if you are packing it in paper bags, then you have to put all the samples in the Ziploc bag, each and every one, because many times when we are getting the samples, that seeds of 2 germplasm which will get mixed, so that is of no use and we have to reject directly. So you have to avoid before sending the sample to the gene bank. Send the seeds to the gene bank as early as possible. When it is harvested, it should be shade dried and should be sent it to gene bank without any treatment. And depending upon the material or germplasm, like if you are going to send the passport data, if you are going to send released variety, then it should accompanied with all the released variety passport data. And similarly for registration of genetic stock, it is the online application. So you have to fill the passport data according to the requisite. And if it is the explored or collected material, then you have to send according to that. And the processing for long-term conservation, what are the sequence which we are doing it? First, receipt of the sample and verification of the documents and registration, allotment of domestic quarantine number, decreased domestic quarantine number, which in the germplasm conservation division, germplasm handling unit is there. So they will be receiving, verifying and allotting the GQ number for further process. After which, if it is sample, it is matched with the original document what you are sending. And if it is satisfied the gene bank standards, then we will clean the seeds and test the seed moisture, test the viability, go for seed drying methods, seed drying and go for packaging, and finally conservation at the national gene banks. Now we will see one by one the procedures and the documents which we require. So as I said, like for explored or collected germplasm, this is the passport data sheet, which it is available in the website. You can see what are all the characters which we required. The collector number, IC number will be indigenous, collection number which we will be providing, common name, botanical name, land race, whether it is cultivar or other thing. Local name is there for land race, then you should write that particular name only. Biological status, type of material, collection, all these data which we require for further process. Coming to the variety, allotment of national identity, this is one, NPPGR is the nodal organization, and we need many times just people, they send their samples only, seed germplasm. For in case of hybrid only, they will send the hybrid, no parental lines. So we need hybrids and parental lines, and along with the variety identification proposals, and in which meeting the variety is identified. That is proceeding of the variety identification meeting, and along with that, the passport data what we are required. Then the third one is registration of trait-specific germplasm, which is otherwise called the registered genetic stocks. This is nowadays online process. So it is user-friendly, and you can go to the NPPGR site and you can find and you can fill it directly. And you have to send the germplasm for registration. to the conservation division, and along with like hybrid means, again there also we need parental lines or any other unique material is there. That also with the document, the publication document, we have to submit it to conservation division for further process. Coming to the seed cleaning, first whatever samples we are getting, we have to check each and every sample to determine whether the seed they have. Sometimes in case of cucurbitaceous vegetable only, they will write sponge gourd, but their material what we are receiving is ridge gourd. Many times, like botanical name or the crop name will be written in different way, and those material we will directly reject it. So, and again we have to wait for, again we are waiting for the intended to reply back whether this passport data is correct, since you are writing crop name is different, botanical name is different, sending the seed is different. So you should avoid all those things. Whatever germ blossom which accompanied with the passport data is final, otherwise material will be rejected. We will check each and every seed packet and see here that opening it in the tray, and tray should contain the labeled one, and we will verify if it is matched with the passport data, then further process will be done. Cleaning of the seeds from fruits and preferably by hand. Remove the debris from the seeds by the use of sieves also we are using. We have different size of seeds. Sometimes like after harvest, they never clean anything. So it contains all the debris. So we will receive it, we are receiving it means then we have to clean everything before sending it to quarantine procedures. And we have got purity boards. There we have to check physically damaged seeds, empty seed, infected seeds, and other seeds, other weed seeds also. Many times we used to get weed seeds, so which can be separated by the use of hand lens or using the purity board, which will be separated and destroyed. See, all the germplasm received by the germplasm handling unit are sent for seed health testing to detect and identify the pest and the pathogens, and pest and the pathogens, weed seeds and other seeds. These are all removed, and in quarantine divisions, like they will salvage it. If it is infected with any pest or diseases, they will salvage it and send it back to germplasm conservation divisions for further processing. So these are all visual observations, and bruchid infestations in legume seeds detected under visual observations. You can see these manually, we can separate it out. Like that, in each and every species, they have reported a lot of insects and fungus which are associated with the germplasm received. Just I would like to say, like medium-term storage facilities also we have got over here, and we are maintaining at 48 degrees centigrade, and the relative humidity is 30 to 35%. Mostly orthodox seeds are stored for 5 to 10 years over here. And here also, like it is working collections and have a higher rate of usage. So we have to go for regular monitoring over it. Other than that, like whatever germplasm which will be rejected from the plant quarantine divisions after the seed health testing, and seeds which not meeting the gene bank standards, maybe of less seed quantity, like cross-pollinated crops, we need 4,000 seeds, but most of the times we are receiving 1,500 seeds also. So sometimes which won't be qualified further for viability test, maybe having 60%. So in that situations, we are keeping the sample in the medium-term storage, and we will request the intender either to multiply freshly and send it after harvest, fresh harvest, or it may be rejected. So till the time we are conserving it in the medium-term storage. And in the medium-term storage, we have got active collection samples which are requested by the different intenders, and we are supplying from medium-term storage, and these are regularly regenerated and supplied for the intended. Coming to the moisture testing, two things we have to remember in the long-term conservation. Two factors. One is the moisture content and the another one is the temperature. So moisture content is the very important factor which determines the rate at which seeds deteriorate and has profound impact on seed longevity in the gene bank. Even small change in moisture content has large effects on seed storage life. Seed aging and loss of germination cannot be stopped but can be minimized by proper seed storage condition. What are all those seed storage conditions? One is the temperature. If low temperature, you can double the life. And if low moisture content, you can double the seed storage life. So according to Harrington's thumb rule on seed storage, this is the famous one. And we are following the same one. For every decrease of 1% seed moisture content, the life of the seed is doubled. And for every decrease of 5 degrees centigrade in seed storage temperature, the life of the seed is doubled. This is applicable where the first rule, 5 to 14% moisture content, you can manipulate and you can increase the seed storage life. The second one is the temperature, which is between 0 to 50 degrees centigrade. So moisture content and the temperature plays a critical role in determining the seed longevity in seed gene bank. How we are determining the seed moisture content? Okay, whatever germplasm what we are receiving, and after passed from the germplasm handling unit to quarantine division and pest-free conservations, we will receive back the germplasm to conservation divisions. Then our mandate is we have to check the initial viability or initial moisture content of the germplasm what we are receiving. There are officially recognized method, that is air woven method. Two type of methods are there. One is the low constant temperature woven method for oily seeds and the high constant temperature woven method for non-oily seeds. For oily seeds, we have to go for slow drying method. There, the temperature is 101 degrees centigrade plus or minus 2 degree, and we have to keep it for the overnight, that is 17 hours. Similarly, the high constant temperature woven method, it is for 130 degrees centigrade plus or minus 2 degree. These are all different for different species. Most of the cereal crops, it is for 1 hour, you have to keep for 130 degrees centigrade. And for the sorghum and the millets crop, it is 2 hours. And for maize, it is 4 hours. According to the ISTA, in the International Seed Testing Association rules, what it says, to promote uniform and complete grinding, observation, moisture estimations, you need grinding. So some of the species what they have listed are Arachis hyboja, Avina, Citrulus, Phagopyrum, Glycine-Max. Most of the seeds which may have larger size, they require uniform grinding. Coarse grinding is there, fine grinding is there. It is according to the ISTA rules, they have mentioned for different species and different crops, different one. Whether it requires fine or it requires coarse grinding. So we have to do, that is necessary, obligatory. Then we have to go for estimation of moisture content. So these are all low constant temperature woven method for defined for brassica oilseed crops. These we have to keep it for 17 hours. High constant method, they have listed so many crops. This may be available in our manual what we have provided. So how we are calculating? We have to calculate the moisture by using this formula. Initial moisture content, we are doing it, and after drying also we have to do the moisture content. It requires maybe 4 to 6 weeks. So we have to take empty bottles. This is very important, crucial one for each and every institute. They want to keep their samples safe, even in the refrigerated conditions. They have to test the moisture. Okay. For this, like empty bottle weight is called M1, and the moisture bottle along with the seed sample. This is also again seed size and the seed sample sizes described in the ISTA rules. You can go for 1 to 2 grams in small seeded crops and for 2 to 4 grams in case of larger seeded crops. Larger seeded crops, again, you need grinding. Okay. Then you have to weigh along with the sample, that is M2. After that, what you have to do, you have to set the woven, whether it is oilseed crop means for 101 for 17 hours, and for cereal crops and other crops means you have to fix it at 130 degrees centigrade and grinder, weigh the sample, open the lid, moisture bottle lid and the seed sample, and kept separately in the oven. Then after the expiry of time, this one, 1 hour or 17 hours, we have to take out the sample, cover the lid in the bottle, keep it in the desiccator, which is having silica gel, then cool it for at least 30 to 45 minutes, after which you have to take the final weight of the moisture bottles. Then you can, using this particular formula, M2 minus M3 divided by M2 minus M1, you will get the moisture percentage. At least you have to do 3 replicates. In case of like gene bank, sometimes like wild and breeding species, we are not able to get so much of seeds in grams. So in that situations, we are going for 2 replications, which lesser number of seeds. We used to count the seed numbers, 25 seeds, 50 seeds, then accordingly, we used to calculate the moisture content and conserved in the National Gene Bank. So moisture content is very important. Now I will hand over the mic to Batma Vigore. So she will explain about the rest of the like germination procedure and other like packaging, drying, and storing it in the National Gene Bank, how we are recording and documenting the germplasm. Okay, thank you very much.
Thank you, ma'am. Thank you.
Good afternoon to everyone. As Dr. Ravi already introduced, myself, Padma Gore, and I'm looking the grain legumes. We are management and conservation of legume genetic resources. And additionally, I'm looking after the assigning the IC numbers to release variety hybrids and their parental lines. So in continuation with the Chitra ma'am, the procedure, after receiving the sample, we go for the germplasm handling unit for the seed health testing, seed moisture content determination, then the seed germination. So we, for assessing the seed viability, we generally use the germination test. For the germination test, we use 400 pure seeds in replicates of 150 or 25 seeds for the cultivated species. And in case of a wild and endangered species where seed quantity is always a constraint, so for that we can reduce or minimize the number of seeds used for the seed viability testing. In that case, we use the 2 replicates each of 25 or 50 seeds for the assessing their viability. And in case of some seeds where seed moisture content is very low, the seeds are very dry, the moisture content is if below 8%, then we have to raise the moisture content of that particular excision. And for that, we allow the, we expose that particular excision to ambient atmosphere for 24 hours so that the moisture content of that particular seeds will go up. So this is about the sample size for the assessing the seed viability. Then the different germination methods are there. Top of the paper method for the small seeded crops like many solanaceous crop, chili, onion, and many cole crops like cabbage, cauliflower, and the millets. We use the top of paper method. And for many leguminous crops where the seeds are medium to large size like chickpea or pigeon pea, we use this between paper method. And the pleated paper method mainly we use for where the seeds require the good amount of moisture or where pleated paper method we mainly use to assess the vigor of that seed or the germplasm. And the larger seed like a castor, groundnut, and beans, we use the sand. And the samples which shows the symptoms or which produces the phytotoxic symptoms on the paper or sand, in that case we use the soil. Here you can see in the picture, this is the top of paper method for the small seeded crops. Very small seeds like this is the tomato seeds we kept in the in the petri plate on the top of paper. So this is the top of paper. For the legumes, where seeds are very large to medium size, we keep the seeds in between the germination paper and then roll the like towel. So this method we also call the roll towel method or the between paper method. This is the pleated papers and the soil, the sand. And after keeping the in the growth media, we keep this growth media along with the seeds in the required temperature and the relative humidity. The temperature for the most of the leguminous crops is 25 to 30 degrees Celsius. So after keeping in the incubator, seed germinator, we assess for their final count and the first count, how many seeds are germinated, how many seeds are normal, abnormal, dead, and hard. And I'm not telling in detail because the separate practical on this assessing the seed viability is there. It will be taken by Dr. Sherry in the tomorrow's practical class. So she will be telling how the seed moisture seed require different temperature and the relative humidity, and so what is the first and the final count for the different species will be taken by Dr. Sherry in the tomorrow's practical class. So next is the seed drying. In the meantime, when we are going for the germination test, moisture test, after taking out the some required amount of the seed, the remaining seed we directly keep in the seed drying room. The different facilities available in the NBPGR are the walk-in seed drying room, seed drying cabinets. Here we have to keep seed in the muslin cloth bag to allow the equilibrium of the moisture content as early as possible. And the temperature and the relative humidity is 15 degrees Celsius temperature and 15% relative humidity is maintained in that walk-in dryer and the seed drying cabinet. So after we keep the seed there for the 4 to 6 weeks till we get the required seeds moisture content, that is 3 to 7%. And in case of wild and weedy seeds where the seed quantity is very less or very precious material, where those material we don't want to share or avoid any loss of that very precious material for wild and weedy species, we use that the cabinet desiccator. These are the cabinet desiccator. Here in the cabinet desiccator, we use the deep blue silica gel for the rapid drying of the seed, and we have to change that silica gel when it turns to blue, because it's absorbed the moisture from the seed and it turns into the pink color. When it turns in the pink color, we have to recharge this silica gel are the reusable. So we have to recharge in oven, and when they turn from pink to blue, then again we can use in this the desiccator. And the saturated salt solution like calcium chloride and zinc chloride we are using for the research purpose, and where the very low moisture content is required, we are using the salt solutions. After we get the required moisture content, that is 3 to 7%, we go for the packaging. For the packaging, we have to hermetically seal our seeds in that aluminum foil packets, and we have to avoid any loss or we have to avoid accessions separate and avoid the mixing of accessions, and prevent absorption of water from the atmosphere after drying. So we have to hermetically seal this aluminum foil packets. This is the sealing machine we have in our NBPGR. The 3 to 4 sealing machines are there. In the meantime, where we are processing for the drying and packaging, we got the IC numbers. So for each accessions, we have to allow the IC number, the national identity number. So we process after verifying the documents for the passport data, the moisture content, the seed quantity, required seed quantities, as already 2,000 for the cell-pollinated crops, 4,000 for the cross-pollinated crops. And in wild and weedy species, this criteria may be reduced up to 500 seed we can conserve in case of wild, weedy, and endangered species, where the seed quantity is getting the required seed quantity is difficult. So after checking all the checklists, we go for the allocating the IC number, and IC number for the release varieties, improved cultivars, for trait-specific germplasm and the collected germplasm are all allocated by the single window from the AKMU, that is Agriculture Knowledge Management Unit. They allocate the IC number, and then we print the barcoding of the barcode labels. Now our NBPGR gene bank is equipped with a barcode, so we got this barcoding labels, and then we paste on that packed material, and after packing we go to the gene bank management information system. Here we create the batch for each and every accessions, and we store each and every accessions according to their location, moisture content, all the details of that whatever we tested, like moisture percentage. Here you can see here we have wrote 7%, what is the germination percent of that particular accessions, and all the details. The most important one is the storage details, where this is the module. As Chitra ma'am told, we have the 12 modules for the different crop curators, and in the modules there are the A to L, A, B, C, D, likewise up to L racks. These are the rotating racks, and on each rack 1, 2, 3, 4, 5 shelves are there. In the shelves we are keeping the baskets, and each basket we keep the accession one by one till the basket get filled. After the filling the basket, we go for the second and third and fourth and so on. So the location of each accession, this is the very important, like module, rack, shelf, and the basket. For the legume crop, I got the module number 9. So this is the accession of black gram. For this particular accession, this is the location. Whenever I require this accessions for particular use for the monitoring, for the supply, for the research or any purpose, I will just search this accession number in our gene bank database system, and I will get this location. That is module 9, rack J, shelf 8, in the number, basket number 48, I will get this accession. The other details like how much number of seeds are there. If I remove some 40 seeds for any test or for the supplying, then I will here update that of how many seeds I remove, I will update in that our gene bank information system, and all other details like passport data sheet and all information we update regularly in our gene bank information database. So these are all our portal, PGR portal, registration and the exchange. All these are the portals. These are detailed given by Dr. Archak already. So I will not give any details about that. And the last one is after packing, after keeping our seed in the long-term storage, the most important one is the distribution of germplasm. So we have the active collections for distribution for research purpose at our 10 regional stations, or 1 MTS here in NBPGR also. But sometimes any accession is not found in any of the active collection or in our regional station, then we go for the base collection. From the base collection, we distribute for the research purpose. In that case, we can supply only 30 to 40 seeds for the research purpose, and along with for distribution of any germplasm, for taking the germplasm from gene bank, we need the material transfer agreement, as Dr. Vandana Teg has already told about material transfer agreement and all these requirements. The more after the supplying seed, we require the more number of seeds to our gene bank back, so that we can enrich our gene bank with a sufficient quantity number of seeds again. So that is all for today, and myself is looking after the grain legumes and Chitra ma'am is vegetables. So if you have any questions about the long-term conservation or for the legumes or vegetables, and additionally I am looking after the released varieties. So if you have any questions regarding that, you can discuss.
Hello, madam. Hello.
Please.
Madam, what will be the temperature for the preservation of the onion seeds?
For the onion seeds,
sir, moisture content we are reducing to 5% and conserving long-term storage is -18°C. So in case if you are doing it in your institute, you cannot bring down, you can bring down to 7% moisture content, and we have to pack it in the aluminum foil packs, which is the important factor. You know? This is 3-layered aluminum foil. But you can get the same specification and reduce the moisture content, store it at your fridge conditions for at least 5 to 7 years. But you have to reduce the moisture content. If you are not reducing, you are storing it at a 10% moisture content, you will lose the viability when compared to storing it in ambient condition. Okay, so
what will be the temperature, storage temperature?
That
storage temperature.
Storage temperature in the gene bank, what we are doing is -18°C. If you don't have facility, you can store it in your medium-term storage condition or in the fridge by reducing the moisture content and packing it in aluminum foil packs.
If we are having the gene bank, can we preserve it at 4°C?
So you are from which institute?
MPKV Rahuri.
So be loud.
MPKV Rahuri, Maharashtra.
Okay.
Okay. So at 4°C is okay.
Yeah, you can.
Storage.
Yeah, medium-term storage is like a 4 to 8°C. But okay, what you have to do, you have to reduce the moisture content.
Reduce the moisture content. Okay.
Yeah.
And for what about groundnut?
Groundnut also 5% moisture content. But in bulk, if you are storing it in medium-term storage, you will keep on opening the modules, okay, MTS. In that situation, so you have to check the relative humidity, what you are maintaining.
Okay, relative humidity, no issue. We are maintaining 30% relative humidity.
Okay.
Okay, humidity has not doubt, but only
okay. Moisture you have to check. That is the important factor, no?
Okay.
Moisture you have to check whether it is 9%, 10% or 5%. 9 or 10 means you can keep it in the cloth bags, so which will equilibrate with the atmospheric conditions what you are having in the MTS. Okay.
For groundnut, we have to preserve the pods or we have to go preserve seeds?
So you can do it both. Pods means you can still prolong the seed storage life. And the seeds means since it is a very thin coated, it may deteriorate fast.
Okay, okay, thank you.
But in the gene bank, we are not storing it a pod. We are removing it like only seeds only we are storing it. But we reduce the moisture content to the level of 5%. Below that also, some in case like 4% also. Oilseed crops, we will get moisture reduction very fast.
Okay. If there any information or bulletin regarding to storage of these oil seeds with NBPGR? Any bulletin?
So standard protocols are there. It is already published. It is in the online. Standard germination protocols and conservation protocols are there. No specific.
Sir, morning I posted, sir, actually in our chat box also. That is the standard seed conservation guidelines.
Okay, it is for all the crops. Definitely.
Yes.
Yeah. It is for all the crops.
All the crops.
We see questions from chat box also. There are some questions.
Okay, thank you.
Ma'am, one question from Muhammad Raja, Dr. Muhammad Raja. They collected old barley seeds, and that means at around 85 years stored seeds in exploration. Is it possible to viable or not?
Yeah, you have to check.
We have to check the viability by the germination test only. We can check always.
Somebody is in online. Please.
Hello, ma'am.
If you have the sufficient quantity of seed, Dr. Raza, you can send to us.
Just a simple test, putting it in the paper or germination seed. They can check that itself, it is viable or not. Otherwise, they can send the seed. We will try with the tetrasolium test, it is viable or not. Maybe it depends upon the conditions, whether it is hot or cold conditions. So we have to check.
Sir, someone is asking, sir, please kindly introduce yourself. Hello. Yes. Any other participants who are interested?
Hello, hello, sir.
Yes.
My Dr. Vinod Kumar, wheat breeder from Pawar Kheda, sir.
Yes.
Hello.
Yes, sir. Audible, sir.
मेरा एक क्वेश्चन था कि हमारे पास एक मीटर है पर उसमें इलेक्ट्रिसिटी रहती है, तो अगर जैसे हम इसमें स्टोर करते हैं, अगर इलेक्ट्रिसिटी डिसकंटिन्यू हो जाती है तो उसका वायबिलिटी पे कोई असर होता है?
सर आपका आवाज कट रहा है।
सर आपका आवाज ठीक से नहीं आ रहा है, आप चैट बॉक्स में भी लिख सकते हैं। सर आपकी आवाज कट रही है, आवाज ठीक नहीं आ रही है।
सर बोलिए बोलिए, या चैट बॉक्स में डालिए सर आप।
अब आवाज आ रही है सर? हेलो।
हाँ बताइए।
बोलिए बोलिए। यस, आ रहा है, आवाज आ रहा है।
हमारे पास में मीडियम टर्म स्टोरेज है हमारे पास हीट के लिए, बट उसमें इलेक्ट्रिसिटी कभी कभी डिसकंटिन्यू हो जाती है, तो क्या उसका असर वायबिलिटी पे होगा, उसके सीट वायबिलिटी पे?
हाउ फ्रीक्वेंटली?
कभी कभी, मतलब जैसे 24 आवर में कभी कभी 5-10 मिनट के लिए या आधे घंटे के लिए, इससे ज्यादा नहीं होती।
नहीं 5-10 मिनट के लिए कुछ हो नहीं सकता। ठीक है सर, वी विल मेंटेन, अदरवाइज वी कैन।
इसमें वायबिलिटी पे असर तो नहीं होगा ना मैडम?
नहीं, 5-10 मिनट के लिए कैसे हो सकता है? इफ इट इज फॉर लाइक 3 days 4 days, और if it is
continuously for hours, then it will affect, definitely it will affect. But अगर 5 minutes के लिए इलेक्ट्रिसिटी जा रहा है तो it cannot affect the viability.
नहीं, 4-5 दिन के लिए तो नहीं होता है, कभी कभी जैसे हो जाता है, कभी आधे घंटे भी चला जाता है, कभी 1 घंटे भी चला जाता है, पर ऐसा कभी बहुत कम होता है। मैक्सिमम टाइम तो 5-10 मिनट के लिए ही होता है। ठीक है, ओके, थैंक यू।
थैंक यू। एनी अदर पार्टिसिपेंट्स? Okay, you can post in chat box also, or otherwise you can send to email also to this training program also. And ma'am, I hope very less doubts are there, ma'am. And
yeah, hopefully they have understand everything, otherwise not understanding it.
No, ma'am. Hopefully it is, ma'am, very good lecture, whatever we are saying.
Since morning they are listening, because Dr. Anjalika also told about the processing and everything.
Yeah, yes.
Since morning only, long-term conservation.
And okay, let the time starts to thanks to our speakers. Ma'am, Chitra ma'am, really it is a wonderful lecture. I highly thankful to you. And Dr. Padma madam, and for you also, it is highly wonderful lecture and very understanding, and some participants also interacted with you, and you concluded the doubts also, and thank you very much, ma'am. And I am requesting all, please kindly switch on videos.
सर सबकी फोटो ले लूं?
हाँ विजय मंडल जी, सब लोग स्विच ऑन कर वीडियोस में रिक्वेस्ट किया। अभी भी कुछ लोग वीडियो ऑन नहीं किया। वीडियो ऑन नहीं किया। मंडल जी?
यस सर। ठीक है सर, हो गया।
हाँ ठीक है, फोटोज ठीक है। थैंक यू वन एंड ऑल।
Thank you.
Yes ma'am.
If you have any queries, anything, you can contact us anytime.
Thank you ma'am, thank you. And thank you participants. for kindly patiently listening for all the lectures. And tomorrow we have here some brilliant lectures are there. That is Dr. A.B. Joshi, who is a wheat breeder from CIMIT, Mexico. And he is working as an Indian representative of CIMIT, and his lecture is there. And another one is Dr. A.K. Roy. He will explain the forest genetic resources and utilization. And next is Dr. Andy Kumar. He will give a lecture on screening PGR for disease resistance. It may useful for your research work. And lastly, in the second session, it is a practical exam is there. That is seed viability assessment by Dr. Sherry Jacob and Dr. G.D. Harish. And tomorrow we will join in at around 9.
Ravi, excuse me.
Yes, ma'am.
Have you received that pre-recorded lecture? Can we play now? Do we have time?
Ma'am, that is now it is not possible, ma'am. It is some downloading issue is there, ma'am.
Okay. Yes. Okay, then later on we'll do.
Yes.
Dr. Martin Van lecture, no?
Okay. Yes, ma'am.
Okay.
And
Martin
tomorrow...
from AVRDC, isn't it?
Yeah, AVRDC. Actually, he was supposed to deliver his lecture, but he got some urgent business through today. So lately he sent some pre-recorded video.
Okay.
But I think some issue is there. So, okay, we'll see it then. We'll play some another time.
Okay, ma'am. Thank you, ma'am. Thank you. And thank you, dear participants. And tomorrow we will meet at 9:30 a.m. And today, Mandal ji, आप
close कर दो.
Meeting close कर दीजिए. Thank you.
Please join before 9:30 by 9:25.
Yes.
Okay, so very senior person.
Tomorrow, please kindly
Dr.
join.
A.K. Joshi from CIMIT, he will be delivering lecture tomorrow. So please be join on time, before time. Okay, thank you.
Thank you all.